Good day, everyone. I'm Dr. John Ward, Director of the Coalition for Global Hepatitis Elimination at the Task Force for Global Health. I also have an affiliation with the National Cancer Institute at the National Institutes of Health. And it's my pleasure to welcome you to this webinar where we'll be exploring the research leading to the development of new therapies for chronic hepatitis B. Next slide, please. This is a series that the coalition puts on with various institutes within NIH, and this one is with the National Institute for Allergies and Infectious Diseases. Just to get oriented to the webinar today, please submit your questions to the Q&A portion of the Zoom that will be on the bottom of your screen. We'll be seeking to answer those along the way, and we'll be having a panel discussion after the end of the presentations where we'll seek to bring some of these questions for broader discussion. The webinar is being recorded. The recording is available on globalhep.org. That's our coalition website. We will also make the video recording will be available in full, as well as for the individual presentations so that people can focus on something that they're particularly interested in. And at globalhep.org, you'll also see the slide sets there. And then we develop a synthesis report touching on the highlights coming from the various presentations. And while you're there, please sign up for our monthly newsletter where you'll get information about upcoming webinars and our other activities as a coalition in the U.S. and around the world. I want to thank the organizing committee at NIH and at the CDC that organizes and plans this webinar series. We've already had a number in the series this year that you can get more information about at globalhep.org. But in particular, I want to thank Betsy Reed Conley, who's the program officer at NCI, who's joins me as the co-chair for the members of this group. I also want to point out that Raj Koshi is a member of this group. He helped put together this webinar series, this webinar that you'll be seeing today. Raj has retired. I believe he'll be joining us today. And so Raj, just wanted to send a personal thank you for organizing this NIAID hosted webinar. Next slide. We've had other NIAID related webinars in the past, particular ones that have gone in depth looking at the hepatitis C vaccine research. Tony Fauci joined that one, but again, the content of these previous webinars are available to you at globalhep.org. Next slide. This is our program for today. You'll be hearing in just a moment with some video remarks from Dr. Jeanne Marrazzo, the current director of NIAID. Then we'll have the presentations that I'll be introducing here in a moment to be looking at, to giving you background information regarding HBV replication and natural history, the various aspects of current therapy for hepatitis B with the current therapies that we have, and then to get into various aspects of new treatments in development for entry inhibitors, capsid assembly modulators, cccDNA inhibitors, immunomodulatory strategies, and then begin to get a public health and patient perspectives on why these new therapies are important for persons living with chronic hepatitis B. Next slide. So let's start off with hearing remarks from Dr. Jeanne Marrazzo, the director of NIAID and NIH.
Hello, everyone. I'm Dr. Jeanne Marrazzo. I am the director of the National Institute of Allergy and Infectious Diseases, and I am really honored to be able to offer some welcoming remarks as you get together for this webinar on the development of new therapies for chronic hepatitis B. I'm really sorry I can't be there in person. I'll be out of the country at the time that you're getting together. But I did want to take the opportunity to say a few words in addition to welcoming you. Hepatitis is an area that I've been very interested in for a long time as an infectious disease physician. And I do not need to remind this group that while we've had many advances in the field, we are still not in a place to claim success when it comes to hepatitis, viral hepatitis in particular. The WHO report, which just came out this year and focused on data from 2022, critically, I think, pointed out how far we still have to go. We still had 1.2 million new hepatitis B infections, most of those occurring in the African region and almost a quarter in Southeast Asia. We still have over 250 million people living with hepatitis B, and half of them are among people 30 to 54 years old. So people who have a lot of life ahead of them and whose lives we'd really like to optimize with good management. And unfortunately, 12% are in children who've acquired the infection maternally or perinatally, and who unfortunately have not had the benefit of vaccination. There's also a big problem with underdiagnosis. 13% of people living with chronic hepatitis B are diagnosed, and only 3% have received antiviral therapy. And this really sets the background for the really unfortunate estimate of over 1 million deaths from hepatitis B virus infection. And that number is not going in the right direction. It's actually increased from 2019. The report also has some very nice data to emphasize what the global burden distribution is of this infection, and I will just point it out to you here. I think a couple of things. Number one, it's everywhere. There is no region of the globe that is spared. And number two, some of the numbers are quite staggering when you look, as I mentioned before, at the 60% plus that are in the African region in blue, and the number in Southeast Asia. So very, very, very large numbers of prevalent cases for us to deal with. When you look at this going forward from the data that we had for 2022, if we do not treat people, we do not get this under control, these numbers, of course, are going to continue to rise. And you can see on this graph, the report has broken out the total number diagnosed in the bottom green, total number eligible for treatment in blue, and the total number treated in pale green. So quite a lot of work to do to try to maintain some sort of control over this. So the report did provide, I think, some very helpful guidance around strategic directions. And I just want to point this one out because this is going to be very relevant, I think, to what you'll be talking about, which is around innovation in advancing the research agenda for viral hepatitis to improve diagnostics and work toward a hepatitis B cure. The research priorities for this response include an improved capacity for in vitro diagnostic medical devices and testing approaches for simplified, timely, and accurate diagnosis of chronic hepatitis B, development of long-acting therapy, formulations for children, given the burden that I mentioned before that are in the pediatric sector, and of course, hepatitis B cure research, going to be very important looking at this over time. So how are we thinking about this? We last updated our strategic plan at NIAID in 2022 for Cure Hepatitis B initiative. And this is thanks to our strategic plan working group. I want to thank them for doing this. There were really three components to this: creating strategies to cure and prevent, understanding the biology of this virus, and then developing tools and resources to make sure that we can affect implementation depending on what we find. So I'll just mention a few initiatives that have come on the heels of this strategic report. In 2019, we had a small business initiative, contract supporting antiviral drugs to cure chronic hepatitis B infection. 2022, an RFA announcing funding for therapeutics for eliminating hepatitis B virus, cccDNA. Talk about that in a minute. 2023 animal models for hepatitis B and C, and then most recently this year, an RFA for multidisciplinary research to accelerate hepatitis B cure in persons living with HIV. So just a reminder of the complex life cycle of the hepatitis B virus as shown there. And it does offer a number of targets, but unfortunately, we have not found the relative magic bullet that we have for hepatitis C. So there are a number of strategies out there in our portfolio that comprise antivirals that we hope can cure chronic hepatitis B infection. Those include development of an RIGI agonist, RNA, preclinical development of HBV capsid assembly modulators, reactive CAMs, which are called RCAMs, a new class of antivirals that target the capsid protein, liver-targeted dihydroquinolonazole molecules, optimization of hepatitis B ribonuclease H inhibitors, and development of star editors using CRISPR technology. So we're moving in some very exciting directions, obviously not quickly enough, hopefully soon enough to make a dent in those projections that I showed you before, but I think a much more robust portfolio than we have had in the last several years. I want to just mention very quickly the portfolio that is specific to therapeutics for eliminating covalently closed circular DNA or cccDNA. Some of those strategies are listed here, and taking a look at a graphic of what that cccDNA looks like. Some of these strategies include high throughput screening, preclinical development of potential inhibitors, chemically ligated guide RNA or LG, RNA-based CRISPR, mentioned that before, peptide nucleic acids that can target this covalently closed circular DNA, perhaps IL-1 beta as a novel agent, and then engineering exosomes for new guide RNA and Cas enzymes. So some exciting areas, lots of promise, but also lots of work to do. A couple of points about the current clinical trial landscape that we are involved with around hepatitis B. I'm sure are aware of our DMID-sponsored clinical trial that is a U01, a first in human phase 1 hepatitis B monoclonal antibody 19 in individuals with chronic HBV infection. This is looking at a potent neutralizing antibody that is directed against the hepatitis B S protein. The goal here is to look at safety, tolerability, and pharmacokinetics of this antibody and to assess the effects on serum hepatitis B surface antigen levels. We are hoping that this will be therapeutic for people living with chronic HBV, and we are actively enrolling in this trial. The AIDS Clinical Trials Group, 5423, is also conducting a phase 2 trial looking at the efficacy of dual combination VIR 2218 and VIR 3434 in people living with chronic HBV with or without HIV who do not have cirrhosis. Obviously, when you move into the cirrhotic phase, you're dealing with a somewhat different situation in many respects. The idea here is a small phase 2 trial to look at the proportion of study volunteers who have a substantial decline defined there in quantitative hepatitis B surface antigen after combination treatment with these two agents at week 24. And these, one is a monoclonal antibody, one is an siRNA targeting the HBV X gene. Target population will be 30. This is in development, but again, I think moving some of these exciting novel agents into people and looking at biomarkers of response is going to really be a very exciting phase. So I'll stop there. A lot more to talk about. I'm sure that you'll have a great meeting. Lots of good discussions. Again, I'm sorry I can't be there, but wish you the best. And please keep up the really critical work you're doing in the field of viral hepatitis. Thanks.
Very
Hello,
much,
everyone.
Jeanne. Yes, thank you very much, Jeannie. John Ward again. I just wanted to thank Jeannie, distance-based, for an excellent set of introductory remarks. Jeannie's been a longtime colleague, and I'm very excited that she's in this new position at NIAID, as I'm sure many of you on this call are as well. Let's now move on over to hear from Dr. Jianming Hu, who's a professor in the Department of Microbiology and Immunology at Pennsylvania State University College of Medicine, where he'll be going and giving us a overview of hepatitis B virus replication in natural history. Jianming?
Okay. Thank you, John. First, I would like to thank John and the organizing committee for inviting me to this interesting seminar or webinar. So I was asked to give a brief introduction to the replication cycle of hepatitis B virus and the natural history of hepatitis B. Next, please. So hepatitis B virus, of course, has been known for a long time. It has, interestingly, multiple types of particles. For virus, of course, it has an intact virion. That's what's depicted on the top right. It has an envelope, and then with viral proteins, 3 viral envelope proteins. And inside the purple layer is the viral capsid, composed of 1 viral protein, the core protein. Inside is the viral DNA with the viral reverse transcriptase or polymerase attached to it. And then also depicted is a protein kinase or PK that's incorporated from the host cell into the virion. Okay. In addition, there are also spheres and filaments sort of depicted at the bottom of the left. So spheres, just about 20 nanometers long, as opposed to the virion, which is about 42 nanometers in diameter. And the filaments is the same diameter as the spheres, except as it says, it's a filament. So both spheres and filaments actually are not intact viruses. They only have envelope proteins. They don't have capsids, and they don't have DNA. So what's also depicted on the right side is a more recently discovered and characterized type of particle we call the empty virus or empty virion. So it has an envelope on the outside, and it has the purple layer, which is the capsid, but it has no DNA and no polymerase. Interestingly, it also has the protein kinase also incorporated from the host cell. Next, please. So this slide depicts sort of a simple diagram of the viral genome. Again, in the innermost circle is the partially double-stranded DNA. We call it relaxed circular DNA. So on the outside, what's called a minor strand, which is complete. In fact, it has a little repeat. On the inner side is so-called the plus strand, which is incomplete as depicted by the dashed line. And so within this very short genome, it's only about 3.2 kb. The virus encodes 4 open reading frames of 4 genes, and from those 4 genes produce 7 viral proteins, most of which I already kind of talked about. For example, the 3 envelope proteins, S, M, and L. The polymerase, which is the longest viral gene, encodes the viral reverse transcriptase, or RT. And then capsid protein I mentioned already, but then on the same open reading frame of the capsid, there's a N-terminal extension. So if the N-terminal extension plus capsid region that consists of the pre-core protein. And then finally, we have the X protein. Okay, so all these proteins sort of are depicted on the outside of the partially double-stranded DNA. And then on the very outside of this diagram, those wavy lines, those are the viral mRNAs. Okay, so the longest one is called 3.5 kb RNA. It's also called the core RNA because it's used to translate to produce the viral capsid protein, as well as the polymerase protein. In addition, it's also called the pre-genomic RNA, or called the pgRNA, because it's the precursor to produce the viral inactive circular DNA by reverse transcription. So in addition to that 3.5 kb RNA, there are also the 2.4 and 2.1 kb RNA. Those two are translated to make the viral L protein and M and S protein, respectively. And then for production of the viral X protein, there's a separate xRNA, the 0.7 kb RNA. So what's also depicted here is all the viral RNAs have the same 3' end. They terminate at the same polymerization site. So indeed, the viral genome is very highly compact. So every open reading frame overlaps with every other open reading frame. And all three viral envelope proteins, for example, are completely embedded within the polymerase gene. And the regulatory sequences are depicted, for example, the enhancer 1, enhancer 2, the basal core promoter or BCP promoter, PRES1 promoter, PRES2, S promoter, and the X promoter. All those are actually overlap with the coding sequences. So it's a very compact genome. Next, please. So this slide depicted the sort of the viral life cycle. So we know a lot about this, but we also don't know quite a bit about this also. Okay, so the virus, again, is an enveloped virus. So it enters the cells through binding to the viral receptor called NTCP. But then afterwards, the entry process is actually not very understood. That's the step 2, depicted with a question mark there. But ultimately, the relaxed circular DNA from the virus is delivered to the host cell nucleus, which is then actually repaired by host DNA repair system to form the so-called covalently closed circular DNA that we already heard about, which is really the basis for viral persistence. And we'll hear more about this later at this webinar. So from cccDNA, the virus then takes advantage of the host RNA polymerase II and transcription factors to produce all those viral RNAs that I just talked about. Those viral RNAs then are exported outside the nucleus to produce the viral proteins. And as I mentioned, the reverse transcriptase protein, the capsid or C protein, the X protein. And the RT and capsid protein then comes together with the pre-genomic RNA that forms the so-called immature nucleocapsids. So that's step 6. So in there, within the immature nucleocapsids, the pgRNA is then reverse transcribed to make a single-strand DNA. Then from the single-strand DNA, making a relaxed circular double-strand DNA. So that's step 7 there. Okay. So once the relaxed circular DNA progeny is made inside the cytoplasm, it can actually recycle back to the nucleus. So that's step 8. So that's the so-called intracellular recycling or re-amplification. So what that does is that in the infected cell, a single nucleus not only just contain the cccDNA come from the infecting virion, but it can also come from the progeny rcDNA de novo made in the cytoplasm. So you can have more than one cccDNA in a infected cell nucleus. Okay. Well, once the viral envelope proteins, again, there's 3 of them, LMS, reaches certain levels, then the mature capsid containing the rcDNA, those like the broken hexagon, are directed to outside the cells, forming virions. So that's step 9 there. So that finished the whole cycle. Okay. So what's also interesting recently people found is there's a low level of pgRNA before it's actually reverse transcribed. So those immature capsids can also be enveloped and secreted as RNA-like virions. So that's step 9 depicted there, okay, with a question mark, okay. And more interestingly, also, like we found that the capsid can form actually an empty capsid inside cells without packaging the RNA or the reverse transcriptase. So those capsids are obviously dead end, but they can also be enveloped by the LMS protein secreted as empty virions. So again, the empty virions have an envelope, has capsid, but has no genome. Okay. So in addition, on the right-hand side, those are depicted spheres and filaments, which I already kind of talked about. So what's important to notice is that spheres and filaments, or surface proteins, can be produced from cccDNA, but in addition, can also be produced from the integrated DNA. Okay? So the integrated DNA comes from double-strand linear viral DNA, which for simplicity I did not depict there. But in the cell, there was always a small minority of double-strand linear DNA, which can be integrated in the host cell genome. So that's step 3A there. Okay? And the integrated DNA can be produced, can be transcribed to make the surface RNA. So that's step 4A. So again, the integrant-derived RNA can be made to produce L and MS protein. So what that says is then the spheres and filaments in the blood or outside cells is not only reflecting cccDNA transcription, but also integrated DNA transcription. So that obviously has important implications for a diagnostics or biomarker. Okay. Next, please. So in addition, actually, could you go back? Sorry. So in step 9C on the very right-hand side, there's the pre-core protein I mentioned, right? So pre-core protein is just slightly longer than the core protein, but the pre-core protein is not usually part of the capsid. Instead, it's secreted outside the cells as a soluble dimeric protein, originally thought to be just this one protein called E antigen after proteolytic processing. But more recently, we and others have identified actually a related protein called pre-C, which is very much related to the E antigen with a slightly different processing. Next, please. So this slide just shows a little bit more detail of the 3 viral envelope proteins. Again, these are produced from the 2.4 and 2.1 kb RNA. So all 3 viral envelope proteins have the same S domain. In fact, the small s protein has only the S domain, but then the M protein, in addition, has the pre-S2 domain, and the L protein then, in addition to that, has the pre-S1 domain. Okay? So, and each one of these have a non-glycosylated form as well as a glycosylated form. So the glycosylated form is called GP, or glycoprotein. Next, please. So here, I just want to bring out some recent structural studies that a collaborator of ours and us published recently through cryo-EM methods. So it's known for a long time, HPV produces a huge amount of spheres and filaments. In fact, those are the basis of first generation HPV vaccine. But we actually did not have a high resolution structure until very recently. So just a couple of years ago, we managed to publish the first high resolution structure of the HBs antigen, the spheres, that's depicted on the right-hand side. It has a quite interesting structure. So one of the most striking thing is, in contrast to what's depicted in the textbooks, these spheres, filaments, and in fact virions, they don't have a typical lipid bilayer. So in fact, HPV is not a classical enveloped virus at all. Okay? So this obviously has implications for virus entry and capsid release from virions, as depicted on the left side. If there is no virion envelope, there would be no viral cell membrane fusion, in contrast to what almost all the reviews or textbooks would depict. So what this says is the viral entry process is going to be quite different from what everybody envisions or depicted so far, based on the structural information we had recently. Next, please. So here, just a little more detail of the viral capsid and pre-core gene. Again, so the capsid is very small, just 21 kD depicted on the top there. It has an NTD, a linker, and a CDD. Okay, and again, the capsid is the major component of the viral capsid. which is also the major component of the complete virion or empty virion depicted on the right-hand side. Okay. So the core protein or P21 is produced from pgRNA, but then the pre-core protein is actually produced from a slightly different RNA called the pre-core mRNA, which is almost the same as the pgRNA, except it's a little bit longer at the 5' end. Because a little bit longer, then it has an additional upstream AUG, which allows the translation of a pre-core protein. It's about 39 minutes longer, the precursor, initially than the core protein. So that's the P25 protein depicted at the bottom. Okay. So from P25, then the signal peptide is removed. So now you have P22. Then from P22, the C terminus is removed, the red part. And now you have the so-called E antigen or P17. So that's the classical E antigen people have been detecting in the blood for decades. But then in addition to that E antigen P17, at the very bottom, there's also protein originally called the P22CR. Recently, we renamed it as a pre-C, which is very similar to the E antigen, except the signal peptide at the terminus is still not removed. So it's very interesting how that protein can be released from inside the cells. Okay. So again, most of the pre-core proteins are not part of the capsid. Instead, they are secreted as dimeric proteins, E antigen and the pre-C protein, as depicted on the right-hand side. Next, please. So here, just a little more structural information. So the capsid structure has been solved quite a long time ago, multiple times indeed. So the capsid basic structure building block is a dimeric. So it's a dimer of a P21. And then 120 dimers then forms a T=4 capsid. So what's depicted here is actually a C-terminally truncated capsid protein, which only has the NTD and the linker region expressed in E. coli from the T=4 capsid. So in the CDD, which is missing from this crystal structure, it has the phosphorylation sites as depicted with those numberings. Unfortunately, it got shifted. The multiple phosphorylation sites, those highlighted serine and threonine residues in both HBV and DHBV. And the CDD is only heavily phosphorylated and dynamically phosphorylated, meaning it's phosphorylated and also dephosphorylated. And it's involved in many functions of the capsid protein. Okay? For example, RNA packaging, DNA synthesis, nuclear organization, and there's probably also involved in regulating assembly, although it's not absolutely required. Okay. Next, please.
Jianming Hu, sorry to interrupt, but you have about 3 more minutes in fairness to the other
Sorry,
speakers.
okay.
So, okay.
Yeah,
So
I'll go much faster. Okay, so I just want
3 minutes. Thanks.
Sure. So the linker originally was thought to be just a spacer. Now actually we have evidence to show it's involved in multiple steps of viral replication. Next, please. Next, please. So here, I just want to say there's a little bit more complications than what I just been describing to you. Although the capsid is usually thought to be composed of a single protein, but recent evidence suggests that pre-core protein, including that P22 and P25, can be a minor component of the capsid protein. And in addition, another truncated capsid protein called HBC182, derived from a spliced RNA, can also be part of the capsid and then actually be important for viral infection. So it just means that the information we have so far maybe not really quite complete as we thought. Okay, next, please. So here I just want to say one word about the X protein. So X protein is involved in regulating viral transcription. The most recent or the accepted version of this is the X protein degrader host factor called SMC5 and 6. So SMC5 and 6 then cannot suppress cccDNA transcription. Okay, next, please. So in terms of cccDNA, I'm sure you'll hear more about this. I just want to go this over quickly. So the relaxed circular DNA is a broken DNA, then it has to be repaired to form cccDNA. Because of time, I'm not going to talk too much because you'll hear more later. Next, please. So this again, I think Dr. Haitao, we're going to talk about this more. I'm going to skip. Next, please. So here, I just want to mention a few words about the virus assembly process. So I mentioned the pgRNA is the template for proteins, the capsid protein or C protein and RT protein. It's so the capsid protein and RT protein can then bind to a RNA signal on the pgRNA called epsilon. So RT binding to epsilon is the first step, and then the capsid binding to the RT and epsilon complex is the second step. So it's a two-step assembly process. Next, please. And it turned out this RNA package signal called epsilon is also the origin of viral reverse transcription, because the viral DNA synthesis actually starts at the 5' end at this epsilon signal. Next, please. And it's through a very unusual mechanism called protein priming, because the primer is actually the reverse transcriptase itself. And we think there are actually still many different ways to tackle this particular step, which remain to be exploited. For example, we've showed you can have a sort of a unusual nucleoside analog like clavidin can inhibit this protein priming step through a very unusual mechanism, non-competitive, and the nucleotide is not incorporated. And I think this is a very interesting mechanism that remains to be exploited for antiviral therapy. Next, please. Okay. So in terms of the capsid, I think we're going to hear more about this. So some of the evidence from our lab suggests the capsid protein is involved in cccDNA regulation. Okay, can increase intracellular amplification, for example, but then block entry. And then, of course, host factors also can regulate entry as well as intracellular amplification. And some of those host factors we think actually determines species specificity. So it has implications for animal model development. And obviously, this has been targeted as antiviral targets. I think Dr. Zlotnick is going to talk about this more. So go ahead, next, please. So anyway, so I think, again, we're not going to talk about the CAM because we're going to hear this again. Next, please.
So if you could summarize, please.
Sure.
We need to move on, please. Go ahead.
Right. So I think, go ahead. I mean, I just want to go to the next slide, please. So this is just a couple of structural slides showing the new information that we have this pocket factor on the viral capsid and how the DNA was organized, that may help us to understand how the rcDNA escapes the capsid to form cccDNA. Next, please. In terms of natural history, I think this is basically says it's a long process that provides opportunities for antiviral therapy, of course, for vaccine to prevent infection, and then we can prevent all the consequences. Next, please. Next, please. Yeah. So in terms of the natural history, I think the most recent identification is we sort of moved over from the previous classification of the 4 different stages, and now we have so-called e-antigen positive infection, e-antigen positive hepatitis, e-antigen negative infection, and e-antigen negative hepatitis with different virological and immunological characteristics. I think this new classification better reflects the immunopathogenesis. Next, please.
Yeah, thank you very much. I think I think just in the interest of time, I think that's a good.
I'll stop here. Thank you.
To move on to Anna Locke to talk about some of the current therapies. Thank you so much. And all of the slide sets will be available on globalhealth.org, so we can get to that. So thank you again for a thorough overview. I'd like to now introduce Dr. Anna Locke, Assistant Dean for Clinical Research, University of Michigan. She's a hepatologist at University of Michigan. I'm sure many on this webinar know her very well. She's been a former president of American Association for Study of Liver Disease, has led a therapeutic guideline development at the global level as well as in the United States. And we're very fortunate to hear her overview of current therapies for hepatitis B. Anna, thank you. You're on mute at the moment.
All right, can you see my slides?
Yes, please go ahead.
Okay. All right, thank you. So I'm going to talk about the current treatment. These are my financial disclosures. Well, we have a very effective treatment for hepatitis B, but we haven't had any new treatment in a long time. We have interferon approved many years ago, and the oral antivirals, the last one of which was 2008. Tenofovir alafenamide is not exactly a new drug, just a new formulation. However, the current drugs actually work pretty well in suppressing virus DNA replication. With 1 year of treatment, you can reduce HBV DNA by about 6 log, which is about 1 million-fold. And entecavir and tenofovir have very similar antiviral efficacy. Interferon does not suppress the virus to the same extent. However, it does have a higher chance of e antigen loss and S antigen loss. Now, one of the very interesting things is that when you suppress the virus, not only do you reduce inflammation, but you can actually also reverse fibrosis, even though we previously thought that fibrosis is irreversible. And this is very nicely demonstrated in the phase 3 tenofovir trial, where they had a lot of patients who had 3 liver biopsies at baseline year 1 and year 5. In red and pink are the patients with baseline cirrhosis. And you can see that at 1 year, this hasn't changed much, but by 5 years with prolonged virus suppression, there is reversal of fibrosis and cirrhosis. Moreover, we also know that antiviral therapy reduces the risk of decompensation. So these are untreated patients. These are patients with suppressed virus. You decrease liver decompensation as well as liver-related mortality. But when it comes to liver cancer, even just suppressing the virus DNA, you reduce that risk, but not completely reduce it unless the patients also achieve S antigen loss, which is a minority. And this is one of the reasons why recent studies are so focused on S antigen loss. Because with the current treatment, the oral antiviral, even with 10 years of treatment, you only get 2-3% of patients clear surface antigen. With interferon, it's a little higher, about 10%, but that's mostly in patients with genotype A. And that's not the genotypes common in Asian countries where the bulk of the hepatitis B patients are. So with current treatment, potent virus suppression can reduce hepatic inflammation and fibrosis, prevent progression to cirrhosis and liver failure, but not completely eliminate the risk of liver cancer. Nonetheless, the NUCs are very safe. Because the current treatment does not eradicate cccDNA or integrated DNA, and there's very low rate of S antigen loss, such that when you stop treatment, virological relapse happens in almost all patients, and therefore long duration of treatment is necessary. And as I've mentioned, the risk of liver cancer persists, even though it is some lower instance. So there is, in contrast to hepatitis C, where we actually achieve a cure with 8 to 12 weeks of therapy. Here we're talking about years and sometimes lifelong treatment, suppression, but not a curative therapy. Therefore, guidelines do not recommend treatment for everyone, at least not at the time of presentation. Treatment is recommended for all patients with cirrhosis, regardless of ALT or DNA. But for those with no cirrhosis, treatment is recommended for those with active disease, which means that they have high DNA as well as elevated liver enzymes, or non-invasive or histological evidence of inflammation or fibrosis. Currently, we do not recommend treating patients with immune tolerant phase, which are the antigen positive, high DNA, but normal liver enzymes, unless they're older patients or they are pregnant, and there we're using it for prevention of mother-to-child transmission. We also are not recommending treatment for antigen negative, inactive patients with low DNA and normal liver enzymes. But there's a lot of controversy as to whether these two groups also deserve to be treated. So as I've mentioned, currently for patients without cirrhosis, treatment is recommended for those in the active phase. So higher DNA as well as elevated liver enzymes, all histological evidence of disease. But then we all recognize that there are a lot of patients in the so-called indeterminate or gray zone. Many people misinterpret the guidelines and say the guidelines do not recommend treatment. Well, if you actually carefully read the guidelines, that's not true, because the guideline recommendation for patients with gray zone is if they have persistently elevated liver enzymes, and the DNA is elevated, and for antigen negative patients, persistently elevated liver enzymes, you rule out other causes of liver disease, you should be considering treatment. Unfortunately, these are really in the notes and not in the bullet points, and therefore a lot of people did not recognize that. Now, in recent years, a lot of people pushing for why are we not treating these patients? They're young and they have high virus level, and we know that persistent presence of antigen and high DNA is associated with increased risk of cirrhosis, liver cancer, and liver-related mortality. And if we treat earlier, maybe we can prevent liver disease from progressing, less integration of HBV DNA, less cancer progression. And while the immune response is poor, it may actually be better when we treat patients when they're young. There are also studies that suggest that even in the immune-tolerant patients, they are at risk of cirrhosis and liver cancer. But in the meta-analysis, when we compare the immune-tolerant untreated patients with the treated non-cirrhotic antigen-positive patients in the active phase, the risk of liver cancer is not increased by not treating them, at least for a short period of time over a matter of years. I think that most of the strongest reason why we're not recommending treating the immune-tolerant patient is that the current treatment does not provide a lot of benefit. So this is a study of patients treated with tenofovir or tenofovir plus emtricitabine for 4 years, antigen positive, high DNA, normal ALT. Even after 4 years, not everyone have suppressed HBV DNA to undetectable, very minimal E seroconversion, no S antigen loss, and every single patient relapsed when treatment was stopped. So if the current treatment, unless you're willing to commit the patients to lifelong treatment, the benefit may be small. And we did the study in the Hepatitis B Research Network funded by the NIAID, including immune-tolerant patients, children, as well as adults. Here, the patients received entecavir lead-in, followed by combination of entecavir and PEG interferon. And we're hoping to achieve antigen loss and sustain viral DNA suppression, but that was only accomplished in 3% of children and none of the adults. So we were very disappointed. And therefore, my stance is that I would not be recommending treatment of patients in the immune-tolerant phase with currently available treatment. Now, that doesn't mean that if we have better treatment, we wouldn't be doing that. The reason for that is that majority of the time, DNA has already been integrated. These patients have minimal inflammation and fibrosis. They're at low risk of cirrhosis and liver cancer during up to 10 years of follow-up. More so, we find that some of the patients, even if untreated, do undergo spontaneous E seroconversion and might have durable remission. And with current treatment, we rarely ever help them with antigen seroconversion. The exceptions, the older patients, pregnant patients, and those who require immunosuppressive therapy, we would be putting them on antiviral. Well, the other group that people have debated whether we should extend treatment would be the patients who are antigen negative with low DNA, the inactive patients, or those in the gray zone. But the first thing is, how do we know if the patient is really in the inactive phase? Because of the fluctuations in virus DNA and liver enzymes. So the guidelines do recommend that you should really monitor patients for 3 times, at least 3 occasions over 12 months, and they're stable with low DNA and normal liver enzyme before you consider them to be truly inactive. Or you add a one-time quantitative S. Low quantitative S is much more likely to be associated with true inactive phase and not someone in the fluctuating phase. Well, why is it that monitoring patients make a difference? because this is a nice study from Taiwan many years ago now. They follow antigen negative patients who start off with low liver enzymes. Now, of course, over time, some of them, the liver enzymes start going up, and these are the patients who are at higher risk of liver-related mortality. However, among those who had persistently normal liver enzymes up to 20-25 years, they really didn't have any liver-related mortality. So the true inactive carriers have pretty good prognosis. But you have to follow them to see if they truly stay in the inactive phase. And this is the tricky part, because a lot of patients do fluctuate. And as I've mentioned, not only do they fluctuate, sometimes when you see them, you can't put them into any bucket. So we call these patients indeterminate phase or gray zone. Now, this is a study which follow a large number of untreated patients, and some of them remain in the indeterminate phase for many, many years. And they realized that, oh, if we don't treat these patients because they are in the indeterminate phase, they do have a higher risk of liver cancer compared to the patients who remain in the inactive phase for many, many years. But they also noticed that really the ones that develop cancer are the older patients. So the older patients, in fact, all the guidelines say when they're in the indeterminate phase and they are older, you should really be considering them for treatment. But there was also another problem with this study, because during the follow-up period of close to 12 years, they were not closely monitoring these patients. These patients had liver enzymes tested less than once a year and DNA tested roughly once every 3 years. So many of these patients might have moved to the more active phase, but were not identified, and treatment was not given in a timely manner. Now, talk about recommendations for starting treatment, but what about stopping? Right now, guidelines would say if the patients have cirrhosis, leave them on treatment, don't rock the boat. But if they don't have cirrhosis and they were antigen positive at the beginning, we treat until yeast cell conversion and undetectable DNA and at least 1 year of consolidation therapy. With antigen negative patients, AASLD guidelines say we should continue treatment until S antigen loss. But the Apostle guidelines and EASL guidelines say, well, we can actually stop treatment in some of these patients because some retrospective studies suggest that taking patients off treatment after 3-4 years might be associated with a higher rate of S antigen loss. Now, initially, those were based on retrospective studies, but there are 3 prospective randomized controlled trials with patients who've been on NUC for several years, and they were randomized to either continue the NUC or to stop treatment. The 2 German studies show that stopping NUC resulted in a higher rate of S antigen loss compared to patients who continued treatment. The Toronto study did not show a difference. Now, the difference between the Toronto study and the German studies was that these were largely Asian patients. 97% of them were Asian patients. And we now recognize, and this is a very nice retrospective study that Harry Jensen put together, more than 1,500 patients, majority of them Asians. What they found is that these patients have been on NUC for 4 or 5 years. If they were Caucasian patients, and the quantitative S is less than 1,000, and you take them off treatment, they have a fairly high rate of S antigen loss. But for Asians, even if the quantitative S is less than 100, the rate of S antigen loss is only 33%. But there were very few patients whose quantitative S dropped to less than 100. Actually, overall in this study, only 5% of Asians achieved S antigen loss when you stop the NUC, but half of them had clinical relapse and had to resume treatment. So this is really not something that should be considered lightly. One has to really balance very carefully. As I've mentioned, with the ability to cure hepatitis C and Hep B being very complicated, many experts say, why do we make life so complicated? Why don't we just treat every surface antigen positive patients? Because after all, even patients who appear to have mild liver disease and low virus are still at risk of disease progression, and we need to constantly monitor them. But some patients don't come back, and we might miss the opportunity of putting them on treatment. But I would counter that. Well, even if you put everyone on treatment, you still need to monitor them, because if you don't monitor them, you wouldn't know that they continue treatment. And if they abruptly stop treatment, you might have a major flare. But also, antiviral treatment reduces the risk of liver cancer, but does not completely eliminate that risk. So for some patients, you might still need to do HCC surveillance. And if the patients don't take the treatment, they're not going to derive a benefit. And as I would show you later, spontaneous remission can occur with hepatitis B, and that's very different from hepatitis C. So this is what we did in the hepatitis B research network study. We started off with patients, majority of them didn't have cirrhosis, and they were not receiving treatment. And we monitored them very closely. These patients, median age of 41, only 1.5% had cirrhosis. Over time, as these patients developed multiple active disease, we started them on treatment. And if you monitor and treat as indicated, you can see that the risk of cirrhosis, liver cancer combined was extremely low. So if you're able to monitor patients, you can have good outcome, and you don't necessarily need to put everyone on treatment right away. Moreover, in this study, we did see patients with e antigen loss. 1/3 of the patients who were initially e antigen positive lost at e antigen. 70% of that was spontaneous, not due to treatment. We had 90 patients who lost S antigen, and almost all of them was spontaneous. So it's not true that everyone will invariably progress. A small proportion of these patients can spontaneously have immune control over the virus and can stay in remission. So my recommendation for hepatitis B is, it's a complex disease, and if we want to provide the best treatment, we as providers need to equip ourselves so that we can understand the disease, and we should participate in shared decision making. That means that we explain and we educate the patients. We present to them the options, and we help them arrive at a personalized decision that is medically appropriate and is embraced by the patient. But because hepatitis B goes through all these ups and downs, we have to constantly revisit the decision and monitor the patient. So a decision that I arrived at with the patient 5 years ago does not mean that that's the same correct decision 5 years later, because the patient is getting older. The virus level might have changed, and the liver disease might have changed. I hope that I've given you a good summary of where we stand now, setting the stage for all the new therapies. Thank you for your attention.
Thank you very much, Anna. That was an excellent overview and a lot of good perspectives just to focus on related to the current therapies. But I think and so we can cycle back to that during the panel discussion. It also just reveals the great demand for expanding treatment to benefit more patients, and that's where the opportunities for new therapies development comes in. So let's move on to hear from Dr. Stephan Urban, who's with the German Center for Infection Research, Heidelberg partner site in Heidelberg, Germany. Stephan has done extensive work on molecular mechanisms of hepatitis B and hepatitis D virus-host interactions. And Stephan and his group developed bulevirtide, the first approved therapy for the treatment of chronic hepatitis D. And we're very fortunate to have Stephan join us today for his remarks. And Stephan, over to you.
Okay, thank you very much. I try to share my Okay, let's start. Go back. Okay, can you see my slides and can you hear me?
Yes.
Yes, we can.
Okay,
yes,
thank you very much. So thanks a lot first for the invitation. Thanks to John, and I want to give a very special thank to Ryan Koshi, who participated in putting that program together. Ryan was really one of the very first people I talked to him on HPV in 1988 when I was a practical student in Munich, and I wish him a very good retirement. I would like to give you an update of entry inhibitors to treat HPV and HDV infection. And let's start. If we look at an entry inhibitor, we look at both viruses in common because they share the envelope protein that you already heard of, the large, small, and middle protein. And the Delta virus uses these envelope proteins. Therefore, Delta virus is very special, not only as a pathogen, replicating by a completely different mechanism. That means that it cannot be treated efficiently with nukes. But it can be treated as HPV may be in the future with an entry inhibitor. And the whole thing that we have to ask ourselves for almost 2 decades is how these viruses can efficiently target liver cells, hepatocytes, go into the liver and do that with a very, very high efficiency. We all know that very low doses of virus is enough to infect a liver for HPV or super infected liver that's already infected with HDV. And we did spend a lot of work in primary human hepatocytes because we were lacking infection system to find out that there are different infectivity determinants, the attachment and fusion, for example. This is the antigenic loop. This means that antibodies that are directed against these loops are not directly interfering with the receptor, but as we know with heparan sulfate proteoglycan, and that the Prius domain in the large envelope protein plays an important role in engaging a receptor that we now know is NTCP, but that was also not known for a very long time because we were lacking the systems to screen for that receptor. If you look at both viruses from a general point of view, they enter via heparan sulfate proteoglycan and NTCP. And then, as you already heard, they deliver they form the cccDNA in the case of HPV or a circular single-stranded RNA in the case of Delta virus. And they can amplify maybe within the cells. That's internal amplification, and an entry inhibitor would not affect these steps. But an entry inhibitor would affect steps that are referred to external amplification. So either super infecting the same cells or then spreading into other uninfected cells. And the question is, of course, to what extent do these external routes contribute to the maintenance of Delta or HPV? Cannot go into detail, but there seems to be a potent role for this mechanism of external amplification, and consequently blocking this effect has therapeutical effects. Now, if you look at entry inhibitors, I just want to mention that, of course, monoclonal antibodies targeting pre-S and preventing infection have already been investigated by Gerlich's lab in 1984. They are very potent inhibitors, antibodies that either directly interfere with pre-S binding to the receptor or with attachment, for example. But they are also like the HPICs, but there are also attachment inhibitors like heparin, suramine, salps, and they interfere with the engagement of heparan sulfate proteoglycan. But the NTCP, sorry, that's a typo, NTCP specific inhibitors like hepcludex or bulevirtide, formerly mercludex, but there are other cyclosporins have been shown, or substrates for NTCP that are transported by that receptor. They could act as entry inhibitors. And for example, bile salts, which are the natural substrates of NTCP, but also other transporters, they have been shown to inhibit infection. But we have very strong differences in the IC50, in the inhibitory activity, and I come to that in a moment why that is, and that's important for the development of entry inhibitor. Let's share with you some thoughts on the hepcludex and some structural details. The hepcludex or bulevirtide has been developed by an infection interference assay. So we wanted to know whether peptidic agents that are derived from the pre-S can interfere with infection. And the bulevirtide is a drug that is not representing completely a natural sequence, but it has been optimized. And you can see if you have these myristylated peptides that just represent this N-terminal part, you can profoundly interfere with infection. This is HPV infection, that's Delta infection in Hepar G cells, but that also works in primary human hepatocytes. And the concentration that you need to interfere with infection are extremely low. That's really amazing. 80 picomolar in primary human hepatocyte. You're in the subnanomolar IC90 levels. And the drug also exclusively target the liver. I show you that in a moment, just a nice picture. And they are specific for NTCP. There's only residual binding to other bile acid transporters in the liver. So that's the consequence of the co-evolution of that HBV virus to its receptor. That's NTCP. It excludes other OATPs, for example, and that's also very important for using these inhibitors as drugs. If you fluorescently label such a molecule like the bulevirtide and you put it onto primary hepatocytes, they need to be differentiated. If they are not differentiated, there's no binding. Receptor is not expressed in HepG2 cells on its own. And the remarkable observation was that if you do a time course, these molecules remain on the surface of hepatocytes, as you can see here, for more than 18 hours. It means the half-life time is very, very long. And that's quite impressive for a receptor because normally it's within seconds or minutes that a receptor is uptaken into the cell. That also may explain the low kinetics of infection of hepatitis B virus. Another interesting finding, if you label radioactively these peptides and inject it into a rat, for example, they target the liver with an enormous efficacy, more than 95% within several minutes. And this peptide is bound to the surface of the hepatocytes on the NTCP. That picture was taken at a time when NTCP was not recognized yet. And if you mutate the peptide with just 2 or 3 amino acids, you get a completely different distribution of that molecule in the organism. We've also done that, of course, during the clinical development in chimpanzees, and you see a similar thing. So at that time, we were asking which molecule is addressed, and we all know now, due to the groundbreaking work also in Willy's laboratory, that it is NTCP, the bile acid transporter, and we have since recently the structure, the cryo-EM structure of that receptor on its own. And what you can see here, it is first an integral transmembrane protein sitting in the membrane, the basolateral membrane of the hepatocyte. It transports bile acid through that tunnel that it can form. And the interesting observation here is that there are obviously 2 bile salt molecules that fit into that may lead to the selection of the bile salts during the transport. Those that are not conjugated, for example, are spit out again. The others are transported. And there is a conformational change driven by a sodium gradient. Here you see the sodium ions that drive that molecule into 2 conformations and allow thereby the bile acid transporting. That's the natural function of that molecule. Now the big question is, how does that bulevirtide, or let's say pre-S NTCP complex looks like? Because that's not only the drug complex to the receptor, it's also, of course, that part of the pre-S domain to the receptor. And in very nice collaboration together with a Swiss group, Kaspar Locher, from the ETH in Zurich, this complex was solved. But you see here is the NTCP in green, and you see the bulevirtide, which on its own is not structured. So it just is a random peptide that does not contain or does not build a structure on its own. And you can separate them into a myristoyl anchor here in pink, and in a black domain, that's the first 20 amino acids, and a string domain, which is then from 20 to or 21 to 48. And if you look here, the myristoyl anchor, which has been resolved in that structure, locates sidewise, intruding the lipid bilayer and positioning the glycine 2 here, the very end terminus, it's the external entrance of that tunnel. And then there's the black domain, and that wedges inside this translocation tunnel where the bile acids are transported through, deeply intruding into the protein and making several specific contacts that irreversibly engage this molecule. And then interestingly, there is that string domain, and we know that's very important for the activity of the bulevirtide, that covers the surface of that block and bridges the extracellular surface of NTCP, clasping that molecule and keeping it in one conformation. And this graph should just show you that binding of the peptide to the receptor, and this is an overlay of the native structure versus the bound structure of NTCP, displaces two core alpha helices or more core alpha helices, and that results in a shift of the panel domain and blocking the bile acid and sodium binding. So the point is that there is no sodium binding anymore. The receptor is irreversibly defunctionalized through that activity. Now, this was all theory. I cannot go into clinical data. The big question arises, of course, what does a bulevirtide entry inhibitor treatment is doing? And I just summarize very shortly the clinical findings here. If you treat patients, and these are 15 patients, this was the MER203 studies, these are delta patients, this is delta RNA. With a monotherapy of Hepcludex alone, or MUB alone, you see a continuous decline in viremia. If you treat with interferon, you also see a decline of viremia, but it's not so profound. It's beneficial, but not so profound. The interesting observation here was that the combination of treatment of PEG interferon with bulevirtide has a very profound synergistic effect. So what we are asking for is, to what extent can an entry inhibitor as a combination therapy lead to maybe a functional cure for delta, and to what extent does a monotherapy help to maintain and control the virus? And this is the results of a recent study that should make us very hopeful for the future. What we had in the 204 study, just to remind you, that has been presented just recently at the EASL meeting. If you continue a co-treatment of PEG interferon with bulevirtide treatment for another 2 years alone, we get about 46% of patients that remained HDV RNA negative for 48 weeks after end of treatment in this 10 milligram bulevirtide group. This is this bar here, whereas the monotherapy is less efficient with curative therapy, and that's end of treatment 48 weeks. So there is a potential for curative treatment of eliminating HDV. This will be discussed in the future, to what extent monotherapy can help and combination therapy. That's not my task to do that, but what I want to end up in the second part is to show you novel approaches to address HBV or HDV entry, because entry inhibition seems to be a very important part of any treatment regimen. And I would like to start to say the problems with bulevirtide. It's effective. It's standard of care in Europe and Russia to treat chronic HDV infection, but it's complex high molecular weight lipopeptide. It requires daily subcutaneous application. It blocks entry efficiently, but also NTCP-mediated bile salt transport. This seems to be asymptomatic, or at least only elevated, but it would be nice to have some different things that do not interfere with bile acid transport. So the aim would be to develop smaller molecules with comparable specific activity, and to develop orally available drugs with high potency and comparable pharmacokinetic behavior. I'll show you one unpublished approach that we followed. So what we did, we used the plug domain I just mentioned, that contains the NTCP essential binding site. But this is much less potent and faster uptaken into hepatocytes. But what we did, we fused them to bile acids. We know that the bile acids interfere with HDV infection with very low efficacy, micromolar range. But what if we fuse the bile acid to that plug with a specific linker? And that's what's shown here. This is the peptidic fraction alone of the small block. The bile acids would be somewhere here, but we can improve the efficacy of these conjugates to the level of Hepcludex or the bulevirtide. They are much smaller, they are chemicals, and they can be maybe, and that's what we investigate, given orally. This is the pharmacokinetics, which is similar to the bulevirtide in mice, at least. There's no clinical trial yet here. They're trying to go for that. But there are other approaches. This is an approach I would like to share with you from Assembly Bio. And what they've done, they screened a library with low molecular weight entry inhibitors in Hep2 NTCP cells for anti-HBV and Delta virus activity. And they had a small molecule here with an IC50 of 29 nanomolar. This is active against all HBV genotypes, similar, which you would expect because it targets as well NTCP, and it's selective towards NTCP. So this is not so efficient against other bile acid transporter. It can interfere with pre-S bindings. So obviously, it targets the same tunnel that I just showed you, and it inhibits also NTCP bile acid transport. This is now in phase 1, is planned. Preclinical studies, I think, performed already, and we will see what comes out of that approach from Assembly Bio. Another approach was an academic approach from Stan van der Graaf. He's an expert in NTCP for a long time, and he cooperated with Johan Nates and also Philipp Möllemann. And they identified an NTCP bile salt uptake inhibitor. They screened for bile salt uptake inhibitors in order to treat cholestatic diseases. And they also looked for the HBV-HDV entry inhibition. And they found a substance shown here that is potent, 28 nanomolar, 60 nanomolar, for HBV or HDV infection. And they could show the antiviral efficacy here in Hep2 NTCP cells for B virus and for the Delta virus. And together with Philipp Möllemann, they looked whether that molecule can be orally delivered to mice, to PHH transplanted mice, and that worked. So this is an oral dose given at day -1. And then HBV infection was initiated, and by 100 milligram per kilogram, they could prevent the infection. So this is the first oral entry inhibitor that is able to prevent infection, whether it is as good as Hep-Gludex and can also prevent the spreading in the liver. We will see, but this is another nice example. Let me go slowly to the end. I would try to show you 2 examples of antibodies. I already mentioned the HBICs that have been used for interrupting the perinatal transmission for a long time. They are HBICs, and they are novel antibodies that are now, and you heard it already from Jeanne at the beginning, from Virology, but there are other companies as well that develop S-specific antibodies that interfere with the heparan sulfate proteoglycans, but there are also pre-S antibodies that are targeting the pre-S domain that are in the region of the Hep-Gludex. And this is some summary of the Vir approach, and the antibody is already called tobevibart. So there is a new name, and there have been recently clinical data presented. This targets a conserved conformational epitope of the S antigenic loop. It blocks intrahepatic HBV and HDV spread in humanized liver mice very efficiently, and the SOLIST phase 2 trial of that antibody showed efficacy on HBS complexation, also HDV serum RNA reduction in patients. And it has already been mentioned that the interesting finding is here that we have a strong synergism with siRNA approaches that lowers the S antigen in hepatitis delta infected patient. I mentioned that there is another antibody, and that there are antibodies against the pre-S. I don't have the time to show you, and I want to sum up with my messages. So entry inhibition is an efficient way to block de novo HBV cccDNA formation, but also HBV cRNA, as I sometimes call it, replication, and also HBV integration. So double-strand linear DNA is also blocked. Virological responses, that's important to understand also for the discussion of primary endpoint, reflect the continuous decline of infected hepatocytes. So it's an indirect antiviral effect. It's not a direct interference with the replication cycle of any of the 2 viruses. Bulevirtide shows efficacy as monotherapy and has curative potential in combination with interferon in HBV-HDV co-infected patients. At least sustained virological responses over 2 years. We will see what happens later on, if there's rebound still possible, if S antigen is not eliminated. NTCP addressing drugs should be selective. They should be irreversibly inactivating the receptor, and they should be not transported. Remember that bile acids are transported within milliseconds through the receptor, and that we have hours where we have an inactivated receptor by these peptidic drugs. Beside bulevirtide, small NTCP targeting molecules are under preclinical development, conjugates, as I show you, orally available molecules, antibodies that address the S domain, like the Vil antibody or the Brias1 domain, show very good antiviral activity in chronic HDV patients. We also see the B trials here, and we will see in which way of combinations they will work. We know that synergistic effects of entry inhibitor are expected, generally seen, with drugs that prevent, for example, the cell division-mediated spread of hepatitis delta virus. This is something you don't address with an entry inhibitor, or with drugs that induce killing of infected cells, so some kind of immune modulatory events. That is, I think, what I would predict to be synergistic to bulevirtide. And the synergistic effect that we see with interferon in chronic hepatitis delta virus should maybe also be elucidated in chronic HPV patients. This is something that we are still lacking too many data on HPV trials. And with that, I would like to thank you for your attention and would like to acknowledge my group, which you can see here, and also people from Walter Meer's group, who did a lot of work on the pharmacokinetics, and of course, Ralf Bartenschlager, who's the head of the department, and for continuous discussion. Thank you very much for your attention.
Thank you very much, Stephan. A great presentation, and just thank you and congratulations for developing the bulevirtide and really, creating the first new treatment for Delta. That's really revolutionized the whole field of screening and linkage to care to identify persons with co-infection for hepatitis B and Delta. So thank you very much, and look forward to you joining the panel discussion here in a moment.
Thank you.
Let me turn now to Dr. Adam Zlotnick, who's the Distinguished Professor of Molecular and Cellular Biochemistry at Indiana University, where he's done a lot of work on the core protein of hepatitis B virus, and will be giving us an overview of capsid assembly modulators. Adam?
Thank you very much. I hope you can hear me.
Yes, I can. We can. Thank you.
And let's make sure I have a pointer. Right. Thanks very much for giving me the opportunity to talk about capsid assembly modulators. Here are my disclosures, and the take-home is that I am an enthusiastic and emphatic advocate for attacking the core protein towards therapeutics and an eventual HPV cure. The 1-minute version of this whole talk is that there are 4 mechanisms of action for CAMs, capsid assembly modulators. 3 of them are based on the fact that the HPV capsid protein, HPC, has structural activities. 1, 2, 3. 1, CAMs are assembly agonists, leading to empty and defective capsids. 2, CAMs bind capsids and can dysregulate disassembly. 3, CAM-induced misassembly can lead to innate immune stimulation and apoptotic destruction of infected cells. HPC also has non-structural activities, and CAMs appear to be able to modulate these as well, making a 4th mechanism of action. The star of our show is the capsid protein, HPC. It's a small dimeric protein with an assembly domain and a basic C terminus. Purified or in cells, it can spontaneously assemble into capsids, predominantly a T=4 120 dimer capsid. And notably, the vast majority of these capsids in cells and in infections are empty. And so when a CAM induces assembly of an empty particle, it's not creating something new, it's following a well-established biological pathway. Jianming went over the viral life cycle. I just want to point out that CAMs and HBC have roles in just about every step of that life cycle. CAMs in particular have roles in disassembly, assembly, and in the nucleus where we have cccDNA. How do CAMs work, particularly as assembly agonists and capsid disruptors? Well, the capsid is built of these dimers, and you can see two dimers here highlighted and here blown up. In the light blue dimer, we have a groove right through over here, and the dark blue dimer fits directly into that groove. This red density over here is actually a CAM molecule. It sits in a pocket in the groove, increases the amount of buried nonpolar surface area, and thus increases the association energy. And it can also modulate the protein-protein angles affecting the assembly products. All known CAMs fit quite nicely into this pocket. Here you see in the apostructure, the groove and the CAM binding pocket. And here you see a dibenzothiazepine, a HAP, a sulfonyl benzamide, and so on. Alternative sites have been proposed for CAMs, and you saw one alternative site identified by at the dimer interface, identified by Zhou Wang, working with Jianming Hu, and also independently by Anya Bachman and Michael Nassel. So what do I mean by inducing assembly? This is purified protein over here, and we can induce assembly simply by adjusting solution conditions. We're looking at light scattering. Big things scatter a lot of light, small things just very little. After we induce assembly, we get an increase in light scattering. The more CAM we have, the more light scattering we get, and the faster it appears. Quantifying these same reactions using the same color scheme by size exclusion chromatography, we see that the more CAM we have, the less dimer we have, and the more capsid and non-capsid polymer. CAMs have been subdivided into CAM-Es that lead to empty capsids and CAM-As that lead to aberrant assembly. In a normal assembly reaction, you come up with a large variety of, actually a large collection of very regular particles, and that regularity is emphasized in these class averages. In the presence of a CAM-A, we should expect more of the same. However, and this is a classic CAM-E, sulfonyl benzamide. However, in class averages, we can see that the vast majority of these particles are actually defective. They are no longer circular. This is nothing nearly as florid as what a CAM-A will do. A CAM-A, like a HAP, favors hexagonal sheets and can lead to huge, monstrous things. Dibenzothiazepines have been identified as CAM-Es and CAM-As, depending on the author, really. But one can see that excess DBT1 leads to tubes and conical particles. So this classification has to be qualified. Cryo-EM shows that CAM-Es lead to defects not easily seen by negative stain, and CAM-A defects are not always apparent when the CAM-A is added at a low dose. So this first mechanism of action, CAM-induced assembly, these are the first generation CAMs. Some of them date back into the late 90s. The two best of these have been shown to have wonderful therapeutic activity, but not be curative. Such molecules are easily discovered and modified, and they can yield normal and aberrant morphology, but the difference between those two morphologies is a little vague. Second mechanism of CAM activity really was developed in this paper by Jan Martin Burke and has since been advanced quite nicely. What was shown was that there's a real difference between the behavior when CAMs are added in the first, immediately after assembly versus after a few days when the infection has had time to establish itself. Under both regimens, the two CAMs investigated were effective at suppressing production of new virus, but the effect of the regimens on production of other biomarkers, notably viral RNA, the E antigen, and the S antigen, were quite different. In an established infection, highlighted in this upper box, neither CAM really had any discernible effect on production of the biomarker. However, when CAM treatment started at about the same time as infection, both CAMs were shown to suppress biomarker formation. Apparently, in mechanism of action 2, CAMs prevent new infection. This effect has to be started at the time of infection, and it apparently works by the ability of the CAMs to melt the infecting virus. And that's really does seem to be the case. Here we're using DBT, one of my favorite molecules, to disrupt capsids. When DBT is added to capsids at low concentration, there's really no effect. But above a certain threshold, capsid disappears and subunits and dimers become the dominant species. I should have noted this is a size exclusion chromatograph. And some work from Assembly Biosciences with a highly active CAM following mechanism of action 2. This is actually work by Naruddin Anshwanawala. The molecule was tested for its ability to disrupt a capsid and expose its DNA to nuclease digestion. At low concentration, not much happens, but above a threshold, CAMs seem to break up the capsid, leading to destruction of viral DNA. The first-gen CAMs had mechanism of action 2, but it wasn't particularly active with the EC50 in the micromolar range. Current generation of CAMs now have 20 nanomolar and lower EC50s for this mechanism of action. Mechanism of action 2, CAM-induced disassembly, is related to assembly. It probably works by stabilizing bad geometry, leading to a rupture of the capsid. And second generation CAMs emphasize both of these mechanisms, disruption and assembly. A third mechanism of action is that CAMs apparently are able to lead to an apoptotic event. Does A stand for apoptosis? This is work that was originally demonstrated by the Alagos group and collaborators. And what they observed is that prolonged treatment with a CAM-A had remarkable effects. The CAM-A led to accumulation of nuclear puncta. And in this mouse model of HBV, a prolonged treatment, well, initial treatment led to a decrease in HBV DNA production. And after a lag phase, there was a loss of S antigen and E antigen. And that loss of these biomarkers was concurrent with the appearance of an ALT flare and correlated with a evidence of apoptosis through a tunnel assay, tunnel staining, and proliferation of uninfected cells. There's a relatively small literature on these apoptotic effects. And in summary, we see that the apoptotic effect is related to aberrant assembly localized to nuclear puncta. Not all CAM-As are created equal, and some seem to be more effective in driving apoptosis than others. The antiviral effect, or the apoptotic effect, requires relatively high levels of HBC, and one can see that without that, one won't have much of an aggregate. And I would anticipate that in integrated genomes where there is no HBC formed, this effect is not going to be very obvious, if at all. The fourth mechanism of activity is the effect of CAMs on cccDNA expression. While it's easy to pick out a starting point for the observation of mechanism, for the first observations of the first three mechanisms, as soon as CAMs started appearing, there seemed to be some evidence for these other activities. Now, earlier, I showed you that CAMs act by binding at a dimer-dimer interface. For CAMs to have this alternative activity, one would expect a CAM to bind to an individual dimer. And recent work from my lab in collaboration with Brian Bothner's shows that when CAMs are added to free dimer, not capsid, it led to an increase in hydrogen deuterium exchange across the molecule. That is, that the hydrogen bonds that are holding alpha helices together are being made or broken at a faster rate in the presence of CAMs than in their absence. Conversely, if you add CAMs to capsids, it slows down the rate of HD exchange. A bit over 10 years ago, Massimo Livrero and collaborators showed that HAP molecules, a CAM-A, were able to affect HBV epigenetics, both in a transiently transfected cell line or in a cell line that is stably transfected and able to accumulate cccDNA. Samples were chipped down and tested for epigenetic markers. Notably, acetylated histone H3 decreased and the amount of bound HBC decreased. My lab, in collaboration with Door Pharma, has identified a subset of molecules that are able to suppress virus production and also S production. And this particular molecule was presented at the HBV meeting in 2021 at DPO4 167. Evidence of CAM effects on cccDNA are present in the literature, sometimes in unexpected places. The goal of this very recent paper by Taverniti et al. Was to examine CAM-A-dependent core aggregation and apoptosis. They used a CAM-A as a control molecule. Interesting, as expected, both the CAM-A and the CAM-A were very good at suppressing viral DNA production, but unexpectedly, both the CAM-A and the CAM-E effectively suppressed S production. For the CAM-A, this is correlated with apoptosis. For the CAM-E, which had no effect on viability, the mechanism is in question. I would suspect it may be a transcription-dependent effect. So mechanism 4, CAMs can act on isolated dimers. CAMs can modulate expression of HBS. CAMs can affect epigenetic markers. The details of this mechanism from CAM to CAM are not known, and any optimization of this mechanism is still at a very primitive level. So in summary, I'm sorry, I'm going a little over time. In summary, There are 4 mechanisms of action: assembly, disassembly, induced apoptosis, and action on cccDNA. CAMs have been optimized for assembly and have completed phase 2 trials with, they've been shown to be therapeutic, but not curative. CAMs have been optimized for disassembly, and these are now in clinical trials. In the future, I look forward to studies of apoptotic induction and action on cccDNA. And thank you very much for your time.
Apologies, and let's turn over to Dr. Haitao to for his presentation, an overview of HBV CCC inhibitors. Thank you very much.
Yeah, thank you, John and Roger, for inviting me to give this overview of finding cccDNA inhibitors. All we can see, we are struggling with cccDNA inhibitors. Okay, this is my disclosure. And Jianming has given a very thorough overview of HBV life cycle. I just wanted to reiterate, the cccDNA basics. So cccDNA is the first viral product made by HBV upon infection. This is a de novo synthesis of cccDNA. And cccDNA is episomal form of HBV DNA genome. It's a mini chromosome and serves as a transcription template. cccDNA itself doesn't replicate. It's only made through conversion of the HBV genomic rcDNA, either through the de novo infection or the intracellular recycling pathway. The cccDNA exists in a low copy number. It's around from 1 to 50 copies per cell. cccDNA is thought to be stable and may survive cell division. We all know that cccDNA plays a key role in the viral persistence and the reactivation of the treatment withdrawal, also responsible for drug resistance to be carried to the progeny virus. So therefore, cccDNA is an undisputed antiviral target for achieving a cure of hepatitis infection. But unfortunately, cccDNA is not only indirectly targeted by the current antivirals. So to target cccDNA, there are 3 major approaches. The first one will be inhibiting cccDNA biosynthesis, maybe together with the nuke, which is a very potent replication inhibitor. So this is blocking the cccDNA formation. And for the pre-existing cccDNA mini chromosome, so either you can reduce the stability of the mini chromosome, or you can silence the cccDNA transcription, because this is the only activity of cccDNA. Considering formation of cccDNA from the rcDNA, so this is a DNA repair process. People can easily understand. The rcDNA can be considered as a damaged DNA because it has the nick gaps and the terminal, peculiarities on the terminus of the rcDNA. So those structures need to be fixed to repair this into a perfect covalently closed circular DNA. So on the bottom of this slide, this is a proposed model for cccDNA formation. It starts from the replicating rcDNA in the cytoplasm. Maybe there is a deproteination step which removes the polymerase from the rcDNA, and it prepares the rcDNA in the nucleus, in the capsid for nuclear transportation. And then in the nucleus, the rcDNA is repaired into the cccDNA by host DNA repair machinery. And in the years of studies from my lab and others' lab, so we have identified a handful of host DNA repair enzymes or factors that involved in cccDNA formation. for example, you know, when the rcDNA get into the nucleus, there's a sensing mechanism like a UVDDB complex or the ATRCHK1 cascade that can recognize the rcDNA in the nucleus and then recruit the host DNA repair enzymes like you know, polymerase exonucleases or endonucleases, you know, to fix the terminate of the rcDNA, or maybe ligate the minor strand first to form this closed minor strand rcDNA, and then you know, the ligase will ligate, you know, the open ends and form the cccDNA at the end. So those DNA repair factors, so a lot of them actually, we have chemical inhibitors available, and some are even used in cancer treatment. So those can be tested for inhibiting cccDNA formation. And using cccDNA formation as an antiviral target has both pros and cons. So to summarize that, so in combination with the nukes, so those cccDNA formation inhibitor, if available, they can block the de novo HBV infection, right? And also can serve as an additional layer of blocking the intracellular cccDNA recycling pathway. And cccDNA itself, if it undergoes DNA damage and can be repaired by host DNA repair machinery, so those formation inhibitor or the DNA repair inhibitors could also block the cccDNA repair. And also the inhibitors of DNA repair enzyme, and as I mentioned previously, used in cancer therapy that can be repurposed for this. But the cons for that is that there's, you know, you're targeting cccDNA formation, there's no direct effect on the pre-existing cccDNA, right? And this most likely is host targeting, and it has uncertain long-term efficacy and toxicity issue. And also from the DNA cccDNA formation studies, we found that there's actually a redundant function among DNA repair factors. So if you block one pathway, another pathway may take over, you know, to repair the cccDNA. And the major issue of targeting cccDNA is the longevity of cccDNA, which is the pre-existing one. They are very stable, and the previous study using the animal model or using the other hepatinovirus actually found that the cccDNA has a very long half-life in vivo. In duck and woodchuck, that could be about 2 months, and in the human, that's around like 6 months, even under nuke treatment. And a study from Marrazzo's lab showed that a complete loss of cccDNA solely by a hepatocyte proliferation cannot be achieved in an immunodeficient mouse model without blocking the viral spread. So if you don't block the de novo infection, the cccDNA still can persist. And this study from Fabian Zolim's lab a long time ago, they measured the cccDNA half-life in the adult treated patients, and they calculate that it may take you know, more than 10 years to completely clear the cccDNA pool in the hepatitis B patients. Maybe this is due to the incomplete inhibition of cccDNA replenishment by you know, a less potent nuke like a adolfir. But even with the entecavir or tenofovir, this may be still an issue, you know, that you cannot completely block the intracellular amplification of cccDNA formation. And later on, you know, a study by scientists in Assembly Biosciences and collaborating with clinicians, they found that by monitoring the dynamic turnover of the nuke-resistant mutants on the serum HBV pgRNA, which is a marker for the cccDNA in the treatment, under the treatment of HBV by nuke drugs. So the half-life of cccDNA pool in the lamivudine or this LDT treated CHP chronic hepatitis B patients actually was calculated at several months. That's shorter than, you know, than the previously thought, but still this is indicate that cccDNA half-life is long. So the cccDNA longevity is maintained by its inherent stability and also the constant replenishment from the replicating rcDNA intracellularly. So both arms should be blocked in order to decrease and ultimately eliminate the cccDNA pool, which is very challenging. And this is the example. So a study from Roche a couple of years ago, they report that they discovered a cccDNA destabilizer. It's called CCCR08. So this compound you can see from the mini circle cccDNA or the stable cell lines all using the HBV infected primary hypothesis. You can see the decrease of cccDNA by the treatment of CCCR08 compound. But the potency need to be further improved because you cannot completely remove the cccDNA. And also there's the mechanism of action is unclear. And also you can from this Sutton blot, you can see the compound also reduce the protein-free rcDNA and the protein-free double-strand linear DNA. So this raises a question, maybe the compound also can target the cccDNA formation or reducing those intermediates for cccDNA formation. And cccDNA, it's a mini chromosome, so it also undergoes the epigenetic regulation for the transcription regulation. So that's including the DNA methylation and the histone post-translational modifications like acetylation or methylation, all these kind of things. And in the cccDNA epigenetic regulation, the viral protein, which is the X protein, plays a key role in maintaining the epigenetic active state of cccDNA for transcription. And from this figure, you can see the X actually actively antagonists many of those restriction factors that can silence the cccDNA, including SETDB1, PRMT1, and the most famous one is the SMAC5 complex. And most recently, we also identify HMGB1 as a silencer of cccDNA, but they all can be antagonized by HBX through different mechanisms. And this shows you that in the absence of X protein, the cccDNA is almost, completely lost the transcription activity. And by this immunostaining, you can also see that, and also by E antigen, measuring E antigen and the pre-core mRNA in the cell. And also without the X, you can see the decrease of the activating histone marker, H3K27 acetylation, and the enrichment of the repressive marker on the cccDNA. So X protein is a very important molecule in maintaining the cccDNA epigenetic status. So when we can compare, the cccDNA destabilization or silencing approach, so here are some thoughts we can discuss. So cccDNA can still be detected in a significant percentage of fully recovered acute hepatitis B patients, despite the presence of active antiviral immunity. And also in the patient, even with surface antigen loss, the cccDNA can still be detected in those patients. And the reactivation of HBV infection in these patients under some circumstance, like immunosuppressive conditions or chemotherapy, is not uncommon. So that's indicating the cccDNA is not completely removed or eliminated from those acute hepatitis B patients with spontaneous clearance. And so it might be difficult to get rid of the very last copy of cccDNA. So silencing that may be the alternative approach. And the only activity of cccDNA is the transcription, as I mentioned previously. So silence the cccDNA transcription activity may be a more realistic or practical treatment option to control hepatitis B. And considering the importance of HBX in the cccDNA transcription, so HBX is the emerging antiviral target for silencing cccDNA. But the HBX is not an enzyme. It functions through protein-protein interactions. So to develop HBX inhibitors to silence the cccDNA. So this is one example from a Japanese group. So they develop an assay to identify compound that block the HBX and the DDB1 interaction, so that it can prevent HBX-mediated degradation of SMAC5/6 complex and then silence the cccDNA. So this is a proof of concept study. You can see they identify a compound called NTZ that can block the HBx and DDB1 interaction. And you can see the rescue of the SMAC5/6 protein in the presence of this compound under HBV infection in the primary hepatocytes. But the potency need to be improved. You can see that this is only a marginal effect on the cccDNA transcription inhibition by the NTZ compound. And recently, we collaborating with Jericho Sciences, which is a North Carolina-based company. They have a HBx binding compound called GS103. So you can also see that the compound block the X-mediated degradation of SMAC6 protein here. And also from the thermal addition study, you can see the compound only blocks the E antigen production after the HBV entry after the entry steps. So that means it targeted the post-entry step, maybe the cccDNA transcription. And this data clearly show you that this compound, without affecting the cccDNA level, it reduced the pre-core mRNA and the ratio between the pre-core mRNA and the cccDNA dose dependently. So this is giving you another example. X can be targeted to silence the cccDNA. And of course, people always think, you can use the gene editing approach to target cccDNA, use the TALEN, CRISPR, or this gene editing or designer nucleases to do that. So, but there's definitely a challenge people always also can think about. The delivery, maybe it's not a bigger problem now because there's LNP, GalNAc, and exome-based delivery, and the liver actually it's probably easier for this kind of a delivery target. But the problem can those, the potency enough to track down the very last copy of cccDNA by those enzymes? And also maybe those approach can select the mutations and the quasi-species based on this sequence. Okay. And the cleavage of the integrated HBV DNA by those designer enzymes may cause the host genome instability because the sequence is the same between the integrated HBV DNA and the cccDNA. And also there's off-target effect could be considered. So, but in terms of the high throughput screening to find the cccDNA inhibitors, so this is still an underexplored area. So the main reason is that we don't have a really reliable and a good marker for monitoring the cccDNA level in the infected cells. So to overcome this, so we have been developing those stable cell line-based system using the E antigen as a marker for cccDNA. So basically, this is the inducible HBV stable cell line. So the integrated HBV DNA will support the replication on the cccDNA formation, but integrated DNA doesn't express the pre-core mRNA and the E antigen. But only when cccDNA is formed, the E antigen can be produced and serving as a marker for cccDNA. And later on, we also add a HA tag into the E antigen region, so which can increase the specificity for this detection. And this is just give you the validation of this system by the old school southern blot, northern blot, and cccDNA. And using this system, which is the first generation of this system, HepD19 cells, we have identified a compound that can inhibit cccDNA formation, which we actually treat the compound at the same time when we induce the HBV replication and cccDNA formation. So this scored this cccDNA formation inhibitor. Presumably, this will reduce cccDNA by reducing the deep protein rcDNA intermediates. And recently, we have slightly modified the screening paradigm by using the induced cells, which already have the pre-existing cccDNA, and then we use the tetracycline 3-TC to prevent the de novo cccDNA formation, and then use the compound to treat the cells and find the compound reduce the E antigen, and then we look back for the cccDNA level and distinguish those inhibitors into the cccDNA transcription inhibitor or the cccDNA destabilizers. For a proof of concept study, so we screened the epigenetic compound library for the cccDNA transcription inhibitors, and we identify MS436. This is the BRD4 inhibitor, which inhibit the BRD4's Haitao activity and the reader activity. And this is just validate how the compound works. It reduces the E antigen, reduces the cccDNA, and also changes the epigenetic status of the cccDNA. And this validate the target, and through the knockdown of BRD4 or use the PROTAC to degrade the BRD4, you can basically recapitulate the same effect of the MS436 compound. But the off-target effect on long-term treatment toxicity need to be further evaluated. And also, surprisingly, we found that when we inhibit the cccDNA transcription by MS436, we also see a slight reduction of the cccDNA level in the treated cells. This is also further demonstrated by a recombinant cccDNA system more clearly. So the implication that cccDNA stability may be influenced by the epigenetic alteration. So, and this is consistent with the previous study, which is the MLN compound that inhibits the SMAC5/6 degradation, and also it reduces the cccDNA transcription, and also you can see the level of the cccDNA reduced. So maybe the cccDNA transcription and stability couple event. So most
we have about one more
actually,
minute, Dr. Gao.
Sure, I will finish.
Please.
So we have further improved that system by replacing the HA tag by a habit tag, which is split luciferase. And using this assay, we can directly lyse the cell by the reagent contains a large bit and measure the habit tag, the pre-core protein in the cell lysate, which is more closely related to cccDNA transcription intracellularly. So and this system we are currently working together with the University of Pittsburgh Drug Discovery Institute to change this into a high throughput platform and for compound screening. So to summarize, so elimination of cccDNA definitely is a holy grail for a complete cure of HBV infection. But silencing and the inactivation of cccDNA is an intermediate goal and perhaps more practical to achieve a functional cure. And of course, inactivation of cccDNA may lead to this destabilization, as some of the studies suggested. A large scale of high throughput screening of cccDNA inhibitor in the cccDNA reporter system and the infection system, also in the presence of nuke or other potent replication inhibitor like the CAM compounds talked by Adam Zlotnick is required. So I just want to stop here and looking forward to discussion. Thank you.
Thank you very, very much. Thorough overview, greatly appreciate it. And let's turn to Dr. Barbara Rehrman. She's the chief of the immunology section, liver disease branch at NIDDK and NIH. And she'll be speaking on immunomodulatory strategies. Barbara?
Thank you. Can you see the slides?
Yes.
Yeah.
Yes. So I would like to thank John and Rajan Koshi for the invitation to speak at this meeting. And like Stephan and John, also congratulate Rajan on his recent retirement and thank him for all of the decades of work and his help to all of the HBV researchers worldwide. His work started at the Max Planck Institute in Munich in Germany and then went over decades at NIH. And I know you're in the audience, Rajan, and I hope you will join the discussion. So I was asked to review immune modulatory strategies. So these are my disclosures. So I'd like to start by asking why cure of chronic hepatitis B is so difficult from an immunological perspective. And it's important to note that the differential disease presentation and outcome really results from differential host-virus interaction due to the different age of infection. So we know, and it's very well established, that HBV infection during adulthood results in an acute, often severe phase of liver inflammation that is attributed to a strong adaptive immune response. The disease is self-limited in almost 95% of cases and results in lifelong cellular and humoral immunity that is protective but not sterilizing because cccDNA persists. So this successful natural immune response is well characterized, biomarkers of protective immunity have been established, and prophylactic vaccines are widely available and effective. In contrast, however, most of the 254 million infected persons worldwide and the many more who are yet undiagnosed have been infected at a completely different age, perinatally or as young children. And there are two important factors that I would like to emphasize. So despite exceedingly high HBV levels in the early years and decades after infection, this does not result in any clinical symptoms, and we do not understand why this virus, while inducing an immune response at that stage, does not really result in any inflammation. And secondly, death due inflammation due to decompensated liver cirrhosis or cancer typically occurs much later than after the reproductive age. And this points to a co-evolution of the virus and the host. And indeed, HBV sequences have been found in 400-year-old preserved mummies, in 3,000 to 7,000-year-old fossils from the Bronze and Neolithic ages. And the entire family of HepatiDNA viridae is thought to have diverged 400 million years ago from non-enveloped DNA viruses in fish. So overall, this shows that there's a long process of co-adaptation and co-evolution of HBV with us, humans, the current hosts. And this is the reason why chronic hepatitis exists and is so difficult to eliminate. So we are simply many, many years behind of the virus. The virus has studied the immune response much earlier than we have. And it's questionable whether we can revert this, but definitely we may be using HBV also as a tool to learn about host immune responses. So it's well known that there are these different phases of chronic HBV infection. As I mentioned initially, there's exceedingly high HBV DNA level and no liver inflammation detectable, no ALT inflammation, and also very high HBs antigen levels. And it's not really clear in the natural history of the disease why this converts to an HBe antigen positive chronic hepatitis phase with changes in HBV DNA levels and inflammation. I think much more immune research on the natural history of HBV, of chronic HBV, is still warranted. What do we aim for? We are aiming for functional cure of chronic hepatitis. This means that HBV DNA serum levels have to be below the level of quantitation, and HBs antigen should be undetectable. Anti-HBs can be present or not present. The loss of HBs antigen alone is associated with beneficial clinical outcome. So these were the criteria established for functional cure at the 2016 AASLD and EASL Treatment Endpoint Conference. More recently, at the 2022 conference, partial cure has been advocated as a next step, as a first intermediate step. And this is the same. Serum HBV DNA have to be below levels of quantification, and that has to persist a minimum of 24 weeks after end of treatment, and after that long term. And HBs antigen levels, if not undetectable for partial cure, can be below the level of 100 international units per ml. The patient has to be HBe antigen negative. So this requires, as we have heard from previous virological talks, a control of HBV cccDNA, the transcriptional template for HBV, and that is established as a mini chromosome in infected patients. This can be achieved via turnover, dilution basically, among progeny of HBV cccDNA containing hepatocytes, but the half-life time of cccDNA is long and the turnover of hepatocytes is small unless there is significant inflammation by immune responses. cytopathic mechanisms, death of HBV cccDNA carrying hepatocytes, non-cytopathic mechanisms of degradation, cytokine-induced, and silencing inactivation of cccDNA are important. And as you can see, many of these factors depend on immune control. In addition to control of HBV cccDNA, we need control of integrated HBV DNA. Seminal studies by Bill Mason have shown that HBV DNA, also non-replicative, integrates throughout all of the chromosomes, except here the Y chromosome at the bottom. And this occurs very early after infection. It already occurs in the E antigen positive phase of viral infection, the first phase, when there's no sign of liver inflammation or clinical disease. Bill Mason has quantitated here the number of hepatocyte clones with integrated HBV DNA. You see that he found 15 to 39 clones in the HBe antigen positive stage in biosamples. And then this increases, and the number increases, and the clone size also increases significantly in the HBe antigen negative phase of infection. And this integrated HBV DNA is considered to be a major source of HBs antigen levels in the blood. So what are the immune modulatory strategies? The first strategy, of course, is to activate the innate immune response. It's well known that HBV does not induce interferon alpha and interferon stimulated genes, but it's a good sign that it is sensitive to them. There's an experiment of nature. We now know that HCV co-infected patients control HBV replication because HCV induces interferon and ISGs. And then if HCV is cleared with direct acting antivirals, this results in an HBV reactivation. So this endogenously produced type 1 interferon response due to HCV infection can control HBV. In vitro, all of these mechanisms here have been shown to be effective antiviral mechanisms, effective also against cccDNA, but this is in vitro and they depend on very high interferon levels. In patients, treatment with pegylated interferon alpha remains the backbone of many of the new immune modulatory combination therapies. The effect is a very rapid induction of interferon-stimulated genes and also a very rapid activation of natural killer cells that are very responsive to interferon because they express high levels of the interferon alpha-beta receptor. And we have shown here in the study that this occurs in both liver and in blood within 6 hours after the first injection of PEG interferon alpha. And there can be a differential response in patients that we have shown to correlate with HBs antigen level decline. And so this very first response is important. And the greater the response, as shown by the patients in red, the better for HBs antigen decline later on through the study. And the patients who do not show an increase here in natural killer cell activation, the black dots, they do have a response, but it occurs later. And this depends on the pharmacokinetics of interferon levels in the blood. And it depends on pre-existing levels of antibodies against PEG, as we have shown here. So HBs antigen loss occurs in 2 to 3% of patients at the end of a 48-week of treatment regimen with pegylated interferon alpha, and then increases long-term after the end of treatment. But it's associated with significant interferon-induced side effects in the patients, and the response can increase if PEG interferon is added on to long-term nucleoside analog therapy. The patient is infected with HBV genotype A, which the majority of patients worldwide is not, unfortunately, and if the pre-treatment level of HBs antigen is low. So how can we otherwise activate innate immune response? A broader activation of innate inflammatory responses, including IL-6, TNF-alpha, and type 1 and type 2 interferon, has been tried with TLR7/8 agonists. Monotherapy with these agonists have minimal clinical efficacy, but I would like to mention one combination therapy study that was presented at the last AASLD meeting. So in patients that received nucleoside analogs, there was an addition of a direct-acting antiviral, an HBV siRNA that blocks S antigen production. And in addition, immune modulators, either a TLR7 agonist or PEG interferon alpha, were added. And as shown here, this increased the percentage of patients that received S antigen loss at the end of treatment, here shown in the light orange, and at the long-term treatment endpoint, week 24, after end of treatment, shown in orange. However, as you can see, the end of treatment time, the week 24 follow-up treatment is about 13% of patients losing S antigen with this combination treatment with the TLR7 agonist. But if you, instead of the TLR7 agonist, include PEG interferon, the response is even greater, and it's 22%. And the response, again, depends on S antigen levels being low prior to treatment at baseline. So what about adaptive immune responses? So we know that in chronic infection, HBV-specific T cells have signatures of exhausted cells. We know this depends on high antigen load and, as shown by Frank Cesare in studies in chimpanzees, on insufficient CD4 T cell help. This sets off an exhaustion program that starts with induction and long-term induction of the activation marker PD-1, CTLA-4, TIM-3, LAG-3, and then impaired effector functions, resulting further down in impaired mitochondrial polarization and accumulation of ROS, and in metabolic and epigenetic changes in the end in loss of some of these cells. The same has been described in HBV-specific B cells that are also dysfunctional in chronic hepatitis B. These cells can now be studied with fluorescently marked BATEs, fluorescently marked HBs and core antigen. And as you can see, as in many other chronic infections, as in HCV and HIV infection, there's an increase in atypical memory B cells in chronic hepatitis B, decrease in classical memory B cells, and there's reduced B cell receptor signaling, production of antiviral cytokines, plasma cell differentiation. So this, both at the T cell level and in the B cell level, has responded to blockade with of PD-1 in vitro and partial recovery of T cell and B cell function. However, in patients, this is quite a different story. We know that HBs antigen-specific T cell responses decline with age. And as you can see on the left, so the percentage of total HBV-specific T cells that respond to S antigen is much reduced once you're over the age of, let's say, 35. And this is specific for HBs antigen-specific T cells, where it's most prominent, as compared to HB core-specific T cells, where we see not such a correlation. So if we then try to block with PD-1 antibodies, this is going to be very difficult because we cannot completely reconstitute. If we try to replace HBV-specific T cells, for example, by bone marrow transplantation from donors who recovered from acute HBV infection into chronically infected recipients, this does work. So you see here, again, an experiment of nature. You see that this patient develops ALT levels increases 28 weeks after bone marrow transplantation, and then seroconverts first to anti-HBe and then to anti-HBs. This is associated with an increase in HB core specific T cell responses. The inverse is also correct. If you transplant an HBV-infected liver into a patient who has anti-HBV immune responses, these immune responses also control HBV in the infected liver. So this is an example that supports the development of specialized, personalized treatment with specific T cells that express very specific HBV-specific T cell receptors or chimeric antigen receptors, as shown in this study here by Tan et al. In a pilot study. This can happen also spontaneously. So we have seen an increase in HBV-specific T cells in patients who seroconvert from HBe antigen positive to anti-HBV positive and HBe antigen negative. And this has also been associated with an increase in functionality of these cells. So here you see in light green graphs the number of HBV-specific T cells when you put them in an in vitro cell culture. And then when you stimulate them with the antigen, you see, as shown by the dark green bars, differential level of expansion of these T cells. So you see greater expansion in those patients who are HBe antigen negative. So this increased T cell number and increased functionality of T cells that we see in HBe antigen negative patients as compared to E antigen positive patients is not associated with increased liver disease. That is important because the liver enzyme levels are lower after seroconversion than in the E antigen positive stage. So this means
I'm sorry, Barbara, can you wrap up, please? Just
yes, I will. So this means
so much.
That an increase in T cell function is not always associated with increased disease inflammation, but rather with increased immune control. Checkpoint inhibitors I mentioned, anti-PD-1 alone resulted here in this study in S antigen loss in a single patient. Newer studies have combined it with seropolitic vaccination with chimpanzee adenovirus prime and MVA boost. And this results in decreases in HBs antigen levels, but the number of patients is still very low. And the HBs antigen levels prior to treatment are also low. Overall, as published by Anna Locke in this very interesting and great recent review article, HBs antigen loss in clinical trials is associated, the strategy is to combine different antiviral treatment and immune modulatory regimens that always include nucleoside analog treatment as a backbone and also often include pegylated interferon alpha. Overall, to summarize, we can replace the inefficient T cell response and antibody response, B cell response in chronic hepatitis B as shown in the left. That is one strategy. There are newer strategies in mice as shown here. We can induce bispecific antibodies, which would make this more as an off-the-shelf treatment, but only shown in mice, not yet in patients. Or we can try to engineer continuous production and better antibodies against anti-HBs antigen, engineer humoral immune responses. Restoration treatments, as shown on the previous slides, include seropolitic vaccination in combination with all of these other new treatment regimens, both antiviral and immune modulatory. From an immunological perspective, I'd like to emphasize that we also need to modulate the immune tolerant environment of the liver, and that any new experimental treatment, if we really want to understand what it does, we also need to monitor immune responses in the liver via fine needle aspirates. Thank you very much.
Thank you very, very much. I wish we had more time to this, but you summarized it very thoroughly for us, so I really greatly appreciate it. Now let's turn to Dr. Sherry Cohen, who's president of the Hepatitis B Foundation. She's also co-chair of the Hepatitis B United Coalition here in the United States. and has been working on hepatitis B related issues for patients living with hepatitis B for many years, and look forward to her thoughts on public health perspectives and patient perspectives as we look for options for functional cures for hepatitis B. Sherry?
Thank you so much, John. Thanks for having me today. And I want to thank all the expert presenters and also Dr. Koshy, who we will greatly miss having you, especially at our International HBV meeting every year. So I'm going to switch it up and talk a little bit about new therapies for hepatitis B from the public health and patient community perspective. Next slide, please. We've heard from expert presenters today on some of the scientific challenges and innovations towards future therapies. And we've heard for many years, we've all been working in the Hep B space for a long time. And in the early 2000s, what we heard was, well, why do we need antivirals for hepatitis B? We have a vaccine. And now what we hear is, why do we need better treatments for hepatitis B? We have antivirals. And I think we've heard very convincing arguments from a scientific perspective about why we need better therapies for the future of hepatitis B. And I'm going to talk a little bit about the challenges and thoughts of people with lived experience. Next slide, please. The data I'm going to share today, we've been collecting since 2019. Primarily, I'm sharing data from a 2020 to 2021 survey. We had about 1,900 individuals participate in this survey, about 1,600 outside the US, 300 inside the US. About 50% of the folks who responded to the survey were on treatment, and about 50% had been diagnosed for more than 10 years. We also, I'm going to share some data that was triangulated from about 150 in-depth interviews and focus groups with about 150 people over the same years. So what we hear, what does it look like to live with chronic hepatitis B infection? We hear about unrelenting, often debilitating chronic fatigue, joint pain, and muscle aches. And these physical symptoms impact productivity, ability to care for family, ability to have a professional life, ability to work, socialize, and attend to necessary tasks. We also hear about somewhat debilitating anxiety and fear and stress, and this is primarily worry about liver cancer and premature death, but also anxiety about being able to afford long-term access and treatment, and anxiety about transmitting to others. We also hear quite a lot about how chronic hepatitis B decreases social interaction and leads to self-isolation and also external isolation. And so this, depending on where people are in their lives, can impact dating and finding a partner, can impact having children. People often are worried or are rejected by family, friends, and coworkers. And we see quite a lot of mental health impact here. Moving on to stigma and discrimination, I think we all have heard the stories of this, but we're also documenting how surface antigen positivity actually leads to sanctioned discrimination in many areas of the world, where people are unable to go to school, unable to get jobs, and unable to immigrate to certain countries. And then, of course, associated with the stigma and shame, and what we hear quite a lot is that people are internalizing that stigma and shame and feel like they're blaming, that they're at blame for hepatitis B infection of themselves and potentially even transmitting to others. And so you can see there's a tremendous impact of living with chronic hepatitis B. Next slide. And this is just a little bit, and you can see that this has been published in a few papers, but to just put a few numbers on it, we created some indices from the 2020 survey, and we created a physical impact, social impact, and worry impact index. And you can see most of the people who responded are greatly physically impacted, but also socially and sort of from anxiety perspectives are impacted as well. We can also see, if you look at the data, that treatment with current antivirals was strongly associated with improvement in all of these indices. However, and I'm going to show you a little bit later, where there was still some residual negative impact. Next slide, please. So currently, there are multiple reasons that people want to take current antiviral treatment, right? So they feel as though they don't have to avoid others. They feel less stigma or less shame. They feel less physical symptoms. However, one of the things that came through from the data was that even when people are on treatment, there are residual quality of life impacts. People know that their liver cancer rate, that their risk of liver cancer does not go down to 0. They know that they can still have, even on antiviral treatment, can still progress. They know that they can potentially still transmit. And there are other considerable treatment challenges, such as cost, side effects, and burden of taking a pill every day for many years. And then I also wanted to quickly just mention that there are many people in the world who do not meet criteria based on many of the different professional society guidelines for current treatment. And those folks are either, what we see from the data is they're either leaving, right? So they're not adhering to follow-up because they're not taking any treatment, or they're trying to find other alternatives to do something positive to manage their hepatitis B. Next slide. So what do people want for future hepatitis B treatment? Next slide, please. Above all, what people want is finite duration therapy that will help them achieve and sustain undetectable levels of surface antigen and undetectable levels of hepatitis B DNA. And they also want to be able to significantly reduce the risk of cirrhosis, liver failure, and liver cancer. And what we hear from people is they want to know that their risk of liver cancer is not necessarily any greater than a person without hepatitis B, and certainly want greater risk reduction than they have today. And then also want to see improved quality of life and an end to stigma and discrimination. And you'll see in a little bit, we're going to talk more about how these things are all intertwined together. Next slide. And this is where we get into some of the details about what people want from future treatment. And you can see on the left, the graph, this is a combination of international and US data. So we ask people to rank their number one reason, their number one wanted outcome for future treatment. And across the board, it was loss of surface antigen internationally. So the top 3 were they wanted loss of surface antigen, they want to be able to stop medication after 6 to 12 months, so finite duration of therapy, and they want decreased risk of liver cancer. But then if you break it down into the US versus international, you can see a couple differences here. So in the US, decreased risk of liver cancer was the number 1 wanted outcome for future therapy. Internationally, it's loss of surface antigen. Although if you look, the top 3 are all the same, the toggles are a little bit different here. And the loss of surface antigen internationally is, of course, intertwined with the fact that surface antigen is what leads to stigma and discrimination. So when people can't get married, when people lose their place in their family, when people can't get jobs and can't immigrate, it is that surface antigen, and that is why loss of surface antigen is so important, especially among people who live in low and middle income countries and are facing discrimination. Next slide. So we can see here that discrimination is certainly a challenge for people with chronic hepatitis B infection, especially in low and middle income countries. We have started a discrimination registry a couple years ago. We've already had almost 600 people complete the registry from about 65 countries. Primarily, we're seeing employment discrimination, but also immigration and education discrimination, as well as healthcare. And this is where functional cure leading to surface antigen loss will play a critical role. However, it also means that we have a challenge now we are being challenged to find a way to change policies in the absence of functional cure. It's great to see new clinical trials coming out that have possibility for functional cure. It's great to see these new innovations, but in the meantime, we need to find a way to make use of the current vaccines and current treatments that we have now so that we can end policy that is discriminatory among people around the world. Next slide. So what do we need? Just to sum up a little bit in terms of future therapeutics for hepatitis B, people want more treatment options. Right now, we know we have interferon, and then we have 2 antivirals, and it's primarily the 2 antivirals that are being used and accessed around the world when treatment is accessible at all. That's not much choice. If you look and try and compare to some other chronic illnesses that are treated with antivirals, there are many choices for antiviral treatments, not just 2. So we need more treatment options better DAAs. We also need more treatment modalities. And I know that there's a lot of excitement in the long-acting antiviral space. And we are actually currently doing a survey right now globally, asking people how they feel with hepatitis B, how they would feel about long-acting antivirals. That could be, that could really help to decrease many of the challenges associated with long-term treatment. Of course, the gold standard would be a cure or functional cure. But then underlying all of it, we need people to have access to affordable and sustainable care and treatment. How do we get there? One of the things that I just wanted to point out today was that, you know, we've seen presentations today in terms of some excellent innovations that could lead to better therapeutics or functional cure. However, over the last 5 years, we've also seen the hepatitis B research space get smaller. First, it got bigger. Now it's getting smaller. We've lost major companies. We've lost small companies just in the past few years and few months. So every time we lose scientists and researchers working in hepatitis B, we lose potential therapeutics for the future. And so we need more funding and prioritization for hepatitis B therapeutics research, and we need more opportunities for emerging scientists to stay in the field. And that, I think, is going to be very important for future therapies for Hep B. Next slide. We also need to make sure that we are meaningfully including the patient community and the people who have lived experience with hepatitis B and the public health community into the preclinical and clinical therapeutic space. We need to make sure that people who have lived experience are included as partners and experts, whether it's in development of treatment guidelines, whether it's in development and implementation of public health programs, or whether it's in preclinical or clinical research, things like design of clinical trials, feedback on clinical trials as well. We want to make sure that patients and the patient community are actually meaningfully represented, not just ticking boxes. And that means working with a number of advocates around the world through community advisory boards, through nonprofit and community organizations who work with, I mean, just at the Hepatitis B Foundation, we work with a few thousand people living with hepatitis B. And there actually are community organizations working around the world who have access to people with lived experience, and folks with lived experience are ready, willing, and excited to come to the table as partners. Next slide. And the WHO, they have a blueprint for this. And so this is from the Global Health Sector Strategy on HIV, viral hepatitis, and STIs. And here it clearly says that in the scientific and clinical community, we should be engaging and supporting people who have viral hepatitis. We need to be doing this around the world, making sure that low and middle income countries and people who are disenfranchised, communities who are disenfranchised, are at the table. Not only will that help to improve uptake of future treatments, it will ensure that people who are highly impacted and communities that are highly impacted will benefit. So from a health equity perspective, we need to make sure that civil society is engaged around the world as we look for new therapeutics. Next slide. I wanted to just end with not me, but the voices of people who are living with hepatitis B. And as you know, July 28th, which was just last week, is World Hepatitis Day. And we have celebrated World Hepatitis Day globally for many years. Last year, we had a declaration published from people living with hepatitis B. And I think we have a very, very short video that we are going to show so that we can hear from folks themselves about what it is they're looking for. I don't think we hear the audio.
Get that.
We want to live healthy and long lives. Care, medication, and liver cancer screening, or our lives may be cut short.
We want to live to our fullest potential, but our personal and professional lives are often limited by stigma and discrimination. We need social support, increased awareness, and removal of any discriminatory laws and practices. We want our doctors to consider the overall impact of hepatitis B and hepatitis delta on our lives and consider outcomes that are important to us.
We want to be at the centre of decision-making for our care, with our preferences and values prioritised. Every year we wait, more of us needlessly lose their lives. We desperately need hepatitis B prioritised, and the time is now.
Thanks so much, Lindsay. And I think that's it for me. So John, I will hand it back over to you. Just one quick, I think I have a last slide in case anyone is interested. On September 11th, we are at the International HBV meeting in Chicago. We are hosting a community forum where we will be hearing from a panel of people who have lived experience and also some of the US experts to talk about progress towards elimination in the US. Thank you.
Thank you very much. It's a great way to end this program, Sherry, because it's really at the end of the day, we're really all about helping people with hepatitis B, and this is a call to action that we all share. I know we're over time, so we don't have a lot of time for discussion. I will call on our speakers here for some closing comments here in just a quick moment, but I did want to just while I had the floor to thank again Raj Koshi for his work on hepatitis over the years, his work as a colleague while I was at CDC and now here at the coalition, being a great partner at NIAID, and I wish him the best in his retirement and look forward to staying in touch with him. I just wanted to see if we're really all about developing these functional cures as well as maximizing the opportunities that we have with our current treatments. I think Jeanne really set us off well by showing the big burden of hepatitis B and what we're trying to reduce, recognizing it's the leading cause of liver cancer, the primary liver cancer, the third leading cause of cancer deaths around the world, really leading into our discussion. So I had a few, just wanted to pose some questions to the group quickly, beginning with, Anna. You really, think there's a real enthusiasm for expanding treatment with our current treatments, but you laid out well sort of the, just sort of the limitations of those therapies. They have real benefits, but then they also have some real limitations. We talk a lot about simplifying treatment, but it seems like, you also laid out a real need to find ways of simplifying monitoring. How frequent do you think people with chronic hepatitis B need to be treated, need to be monitored to capture those that are, changing their natural history so that treatment would be warranted, number one, and number two, do you think we should have more age-based recommendations to start treatment, given some of the reasons you raised in your talk?
So monitoring, obviously, the more frequent, the better. On the other hand, we also have to be practical because this is not about monitoring for a year or two. You're talking about monitoring for years and decades. And I think the first couple of years is the most important because you really want to establish whether this patient has active disease, quiescent disease, or in between. Once we have established that the patient is in the inactive phase, you can do monitoring once a year. And I think that once a year is about as infrequent as it should be, because otherwise you would miss those opportunities to start them on treatment. In terms of how liberal do we make the guidelines, if you have Hep C treatment, something similar to Hep C treatment, it's just a no-brainer, right? You get a cure, Oral treatment, 8 to 12 weeks, very safe. It's just limited by cost. But whenever you're talking about many years of treatment, it's not just that you start the patient on treatment. If you don't have the patient's commitment to stay on treatment and stay with the program, you're kidding yourself that you're doing the patient good. You give them a prescription and you say bye, and you don't have the mechanism of following them. And that's where I sort of have some concerns about, let's just treat everyone, because we don't have the capability of monitoring everyone. And obviously, the more educated patients are more likely to be adherent than others.
Thank you. Thank you very much. Let me turn it over to Stephan. He can have some remarks, Stephan, just from the other presentations. But you raised your efforts to develop a point of care test for Delta infection, and what are your prospects for that? Is it an antibody test or a virologic point of care test? What are you looking to develop, please?
Mm-hmm. Yeah, thanks for the question. I think the under-testing of Delta is not only a problem in, for example, African countries, it's also a problem in the US or in some developed countries. Yeah, and we're really lacking information on epidemiology, and we have to identify by a cheap method Delta, probably best together with HBs antigen. So we've developed a pan-genotypic test. And we are in the process now of getting that to the market, which will take a little bit time. It's a blood test for self-testing in principle. And we hope that we will have that soon. And I think it's very important, not for retract testing or reflex testing, for example. You can, of course, use that as well, but for really testing in field, yeah, and and we will see, and of course we will keep you updated for the testing. And the final goal is then also to combine it with S antigen, so that you have antibodies, IgGs against Delta and S antigen in one test. That would be a good first test. Of course, has to then to be evaluated whether Delta RNA is there in order to treat patients if there's available drugs until that time.
Excellent. Thank you. Look forward to see how that development progresses. Let me turn it over to Adam. And just invite any closing comments you'd like to make, but just in terms of capsid assembly modulators and sort of getting over to Sherry's remark about the sort of the universe of companies and individuals working on new therapies. What's your view of the prospects of developing therapies that will actually be available for patient use? Is that still a long-term target, or you think it's one that could be realized in the next 3 to 5 years?
I wish I had a crystal ball. There's 3 separate questions you're asking all at once. The economics of companies working on HBV and the good fortune to come up with the right hypothesis and then executing on that hypothesis. A problem is there are fewer and fewer, as Sherry points out, there are fewer and fewer companies working in the area and willing to invest in the area. Companies seem to be fairly gun shy, and similarly, study section panels tend to be very conservative. We need people and companies willing to take on high-risk, high-return projects. 3 to 5 years. There are some advances in recent history with the core protein directed molecules that have me incredibly excited. The apoptotic effect of some molecules, as a for example. But unless we execute and invest in the preclinical work, we'll never bring it to the clinic.
That's right. Thank you. I sort of have the same question for Haitao and for Barbara. So if you're in the area of therapeutics you were touching on, what's your crystal ball as far as drug development leading to patient care with new therapies?
I think the immunologists already have the crystal ball if they understand how the acute hepatitis B is cured, right? Yeah, I think the using pharmacological intervention with cccDNA by small molecule remains very challenging. Everything is preclinical now, and even just in the lab is under cooking. So cccDNA is really not a very easy target because it's not a single molecule. It's a big complex, and most likely a lot of the the antivirus targeting cccDNA will be the host targeting. So there's a lot of issues, but I do think if we put more effort on that and do a really large scale high throughput screening for cccDNA, we will find something that can open new doors, like Adam's company called Door, right? We can open new doors to looking for cccDNA inhibitors. And also those information will be greatly helpful to understand the cccDNA biology and further develop targets for targeting the cccDNA.
That's great. I was really interested in your HBX interaction. I think that's really sort of a new area as well. Barbara, what are your thoughts, please? Oh, sorry. Go ahead.
I again would like to make the point that I think we cannot expect to restore the immune response in chronic patients to the level that those that spontaneously recovered had, because in the chronic patients, they never had such an immune response. The priming, the generation of the immune response in the neonatal or early life stage is completely different. So the easiest thing would be to have sterilizing immunity or to have antivirals that completely eliminate cccDNA. Otherwise, we need to find something that maintains an immune response in chronic infection to a level that controls. So if you transfer the bone marrow, that works. But otherwise, how do you maintain an immune response to that level so it continuously controls cccDNA? That's, I think, difficult. So I would definitely, crystal ball means we need antivirals too, and we need a combination of everything.
Thank you. Very good. And Jianming Hu, you started us off with an overview of the replicative cycle, et cetera. So after hearing these talks, do you have any other thoughts you'd like to share?
I think I would just like to concur with Barbara just mentioned. I think we do need everything. In fact, I doubt with the nukes alone we can ever achieve 100% inhibition of even just replication, let alone the pre-existing cccDNA. Okay, so if we could have combination of inhibitors, that maybe we can have a deeper suppression of replication, which hopefully can turn into at least a blocking of new cccDNA production. Maybe let the host, the cell, take care of the pre-existing cccDNA, or the immunologists can figure out a way to target the infected cells and somehow get rid of the infected cells. And beyond that, I think I agree we need more research and more efforts. Thank you.
Thank you. Well, one of the byproducts, I have to say, of these webinars is to keep hepatitis B related research in front of NIH, to let them know that people are interested in this area of research, and just help raise awareness and the education level of what has been achieved to date. It's great to hear from Jeanne her interest in hepatitis B, and I think that's a very good sign for us to continue to see how we can expand this research portfolio in the future, and hopefully, working with other institutes like NIAID, of course, as well in that regard. So I think I'll close it off from now. I appreciate everybody staying over a little bit late. It's an excellent set of presentations. All of this information will be available to others on our website at globalhep.org. I look forward to working with all of you in the future to expand access to the current treatments that we have, the brand new treatments we have, such as bulevirtide, and then look to see how we can how this drug development we've been talking about over the last several hours can translate into new therapeutic options for patients living with chronic hepatitis B. Thank you very much again.